chloroplast. 84 languages. Structure of a typical higher plant chloroplast. The green chlorophyll is contained in stacks of disc-like thylakoids. A chloroplast, /Klrplast, PLST /1, 2, is a type of organelle known as a plastide that conducts photosynthesis mostly in plant and algal cells. Chloroplasts have a high concentration of chlorophyll pigments which capture the energy from sunlight and convert it to chemical energy and release oxygen. The chemical energy created is then used to make sugar and other organic molecules from carbon dioxide in a process called the Calvin cycle. Chloroplasts carry out a number of other functions, including fatty acid synthesis, amino acid synthesis, and the immune response in plants. The number of chloroplasts per cell varies from 1, in some unicellular algae, up to 100 in plants like Arabidopsis and wheat. Chloroplasts are highly dynamic, they circulate and are moved around within cells. Their behavior is strongly influenced by environmental factors like light, color and intensity. Chloroplasts cannot be made anew by the plant cell and must be inherited by each daughter cell during cell division, which is thought to be inherited from their ancestor, a photosynthetic cyanobacterium that was engulfed by an early eukaryotic cell. 3. Chloroplasts evolved from an ancient cyanobacterium that was engulfed by an early eukaryotic cell. Because of their endosymbiotic origins, chloroplasts, like mitochondria, contain their own DNA separate from the cell nucleus. With one exception, the amoeboid Paulinella chromatophora, all chloroplasts can be traced back to a single endosymbiotic event. Despite this, chloroplasts can be found in extremely diverse organisms that are not directly related to each other, a consequence of many secondary and even tertiary endosymbiotic events. Discovery and etymology. Problems donating. Other ways to give frequently asked questions. We never sell your information. By submitting, you are agreeing to our donor privacy policy. The Wikimedia Foundation is a non-profit, tax-exempt organization. If you make a recurring donation, you will be debited by the Wikimedia Foundation until you notify us to stop. We'll send you an email which will include a link to easy cancellation instructions. I already donated. How often would you like to donate? One time give monthly. Please select an amount, USD. The average donation in the United States is around $13. $2.75, $10, $15. $25, $50, $75. $100 other. Please select a payment method. Bank transfer, ACH. Maybe later. 1. Suzo RCN. 2. 3. Continue. Wikipedia still can't be sold. The 17th of October, an important update for readers in the United States. You deserve an explanation, so please don't skip this 1-minute read. We're sorry to interrupt, but it's Thursday, the 17th of October, and this message will be up for only a few hours. We ask you to react on the number of times you visited Wikipedia in the past year and if you're able to give $2.75 to the Wikimedia Foundation. If everyone reading this gave just $2.75, we'd hit our goal in a few hours. In the age of AI, access to variable facts is crucial. Wikipedia is at the heart of online information, powering everything from your personal searches to emerging AI technologies. Your gift strengthens the knowledge of today and tomorrow. Just 2% of our readers donate, so if you have given in the past and Wikipedia still provides you with $2.75 worth of knowledge, kindly donate today. If you are undecided, remember that any contribution helps, whether it's $2.75 or $25. Proud host of Wikipedia and its sister sites. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Chloroplasts, containing thylakoids, visible in the cells of Tychostomum capillaria, a type of moss. Primary endosymbiosis. A eukaryote with mitochondria engulfed a cyanobacterium. In an event of serial primary endosymbiosis, creating a lineage of cells with both organelles. 17. Secondary endosymbiosis consisted of a eukaryotic alga being engulfed by another eukaryote, forming a chloroplast with 3 or 4 membranes. The first definitive description of a chloroplast, chlorophyll cornin, grain of chlorophyll, was given by Hugo von Moll in 1837 as discrete bodies within the green plant cell. 4. In 1883, Andreas Franz Wilhelm Schimper named these bodies as chloroplastids, chloroplastidin. 5. In 1884, Edouard Strasburger adopted the term chloroplasts, chloroplassin. 6, 7, 8. The word chloroplast is derived from the Greek words chloros, which means green, and plasts, which means the one who forms. 9. Endosymbiotic origin of chloroplasts. See also, cyanobacteria and semiogenesis. Chloroplasts are one of many types of organelles in photosynthetic eukaryotic cells. They evolved from cyanobacteria through a process called organellogenesis. 10. Cyanobacteria are a diverse phylum of gram-negative bacteria capable of carrying out oxygenic photosynthesis. Like chloroplasts, they have thylakoids. 11. The thylakoid membranes contain photosynthetic pigments, including chlorophyll. A. 12, 13. This origin of chloroplasts was first suggested by the Russian biologist Konstantin Merezhkovsky in 1905, 14, after Andreas Franz Wilhelm Schimper observed in 1883 that chloroplasts closely resemble cyanobacteria. 5. Chloroplasts are only found in plants, algae, 15. And some species of the amoeboid Paulinella. 16. Mitochondria are thought to have come from a similar endosymbiosis event, where an aerobic prokaryote was engulfed. 17. Primary endosymbiosis. Approximately 2 billion years ago, 18, 19, 20, a free-living cyanobacterium entered an early eukaryotic cell, either as food or as an internal parasite, 17, but managed to escape the phagocytic vacuole it was contained in and persist inside the cell. 12. This event is called endosymbiosis, or cell living inside another cell with a mutual benefit for both. The external cell is commonly referred to as the host while the internal cell is called the endosymbiont. 17. The engulfed cyanobacteria provided an advantage to the host by providing sugar from photosynthesis. 17. Over time, the cyanobacterium was assimilated, and many of its genes were lost or transferred to the nucleus of the host. 21. Some of the cyanobacterial proteins were then synthesized by host cell and imported back into the chloroplast, formerly the cyanobacterium, allowing the host to control the chloroplast. 21, 22. Chloroplasts which can be traced back directly to a cyanobacterial ancestor, i.e. without a subsequent endosymbiotic event, are known as primary plastids. Plastid in this context means almost the same thing as chloroplast. 17. 23. Chloroplasts that can be traced back to another photosynthetic eukaryotic endosymbiont are called secondary plastids or tertiary plastids, discussed below. Whether primary chloroplasts came from a single endosymbiotic event or multiple independent engulfments across various eukaryotic lineages was long debated. It is now generally held that with one exception, the amoeboid Paulinella chromatophora, chloroplasts arose from a single endosymbiotic event around 2 billion years ago. And these chloroplasts all share a single ancestor. 19. It has been proposed this the closest living relative of the ancestral engulfed cyanobacterium is Gliomargarita lithophora. 24, 25, 26. Separately, somewhere about 91-40 million years ago, this process happened again in the amoeboid Paulinella with a cyanobacterium in the genus Prochlorococcus. This independently evolved chloroplast is often called a chromatophore instead of a chloroplast. 27. Note 1. Chloroplasts are believed to have arisen after mitochondria, since all eukaryotes contain mitochondria, but not all have chloroplasts. 17, 28. This is called serial endosymbiosis, where an early eukaryote engulfed the mitochondrion ancestor, and then descendants of it then. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The glaucophyte Cyanophora Paradoxa with 2 chloroplasts in the process of dividing engulfed the chloroplast ancestor, creating a cell with both chloroplasts and mitochondria. 17. Secondary and tertiary endosymbiosis. Many other organisms obtained chloroplasts from the primary chloroplast lineages through secondary endosymbiosis, engulfing a red or green alga with a primary chloroplast. These chloroplasts are known as secondary plastids. 23. As a result of the secondary endosymbiotic event, secondary chloroplasts have additional membranes outside of the original 2 in primary chloroplasts. 29. In secondary plastids, typically only the chloroplast, and sometimes its cell membrane and nucleus remain, forming a chloroplast with 3 or 4 membranes. 32. The 2 cyanobacterial membranes, sometimes the eaten alga's cell membrane, and the phagosomal vacuole from the host's cell membrane. 29. The genes in the phagocytosed eukaryote's nucleus are often transferred to the secondary host's nucleus. 29. Cryptomonids and chlorarachniophytes retain the phagocytosed eukaryote's nucleus, an object called a nucleomorph. 29. Located between the second and third membranes of the chloroplast. 12, 22. All secondary chloroplasts come from green and red algae. No secondary chloroplasts from glaucophytes have been observed, probably because glaucophytes are relatively rare in nature, making them less likely to have been taken up by another eukaryote. 29. Still other organisms, including the dinoflagellates Carlodinium and Carinea, obtained chloroplasts by engulfing an organism with a secondary plastide. These are called tertiary plastids. 23. Primary chloroplast lineages. All primary chloroplasts belong to one of four chloroplast lineages: the glaucophyte chloroplast lineage, the rhodophyte, red, chloroplast lineage, and the chloroplastidon, green, chloroplast lineage, the amoeboid Paulinella chromatophora lineage. 33. The glaucophyte, rhodophyte, and chloroplastidian lineages are all descended from the same ancestral endosymbiotic event. 29. Glaucophyte chloroplasts. See also, glaucophyte. The glaucophyte chloroplast group is the smallest of the three primary chloroplast lineages, as there are only 25 described glaucophyte species. 34. Glaucophytes diverged first before the red and green chloroplast lineages diverged. 35. Because of this, they are sometimes considered intermediates between cyanobacteria and the red and green chloroplasts. 36. This early divergence is supported by both phylogenetic studies and physical features present in glaucophyte chloroplasts and cyanobacteria, but not the red and green chloroplasts. First, glaucophyte chloroplasts have a peptidoglycan wall, a type of cell wall otherwise only in bacteria, including cyanobacteria. Note 2. Second, glaucophyte chloroplasts contain concentric unstacked thylakoids which surround a carboxysmon-icosahedral structure that contains the enzyme rubisco responsible for carbon fixation. Third, starch created by the chloroplast is collected outside the chloroplast. 37. Additionally, like cyanobacteria, both glaucophyte and rhodophyte thylakoids are studded with light-collecting structures called phocobolisms. Excavator. Possible cladogram of chloroplast. Evolution, 29, 30, 31. Circles represent endosymbiotic events. For clarity, dinophyte tertiary endosymbioses and many non-photosynthetic lineages have been omitted. It is now established that chromalveolator is paraphyletic to Rhizoria. 31. Edit. Land plants. Euglonophyta, Rhodophyta, Chromalveolator, Rhizoria, Paulinella, Chlorarachniophyta, Haptophyta, Cryptophyta, Heterocontophyta, Dinoflagellator, Apicampal, Z. Ciliate, Cyanobacteria, Glaucophyta, green algae, Archaeplastida. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Diversity of red algae clockwise from top left, Bornetia secundiflora. Peysonelia squamaria, Cyanidium, Laurentia, Calophyllus. Lacinata. Red algal chloroplasts are characterized by phocobilin pigments which often give them their reddish color. Diversity of green algae clockwise from top left, Cenodesmus. Micrasterias, Hydrodictyon, Volvox, Stygioclonium. Green algal chloroplasts are characterized by their pigments chlorophylla and chlorophyll B which give them their green color. Rhodophyta, red chloroplasts. See also, red algae. The rhodophyte, or red algae, group is a large and diverse lineage. 29. Rhodophyte chloroplasts are also called rhodoplasts. 23, literally red. Chloroplasts. 38. Rhodoplasts have a double membrane with an intermembrane space and phycobilin pigments organized into phycobilisms on the thylakoid membranes, preventing their thylakoids from stacking. 12. Some contain pyrenoids. 23. Rhodoplasts have chlorophyll and phycobilins. 31. For photosynthetic pigments, the phycobilin phycoerythrin is responsible for giving many red algae their distinctive red color. 39. However, since they also contain the blue, green chlorophyll and other pigments, many are reddish to purple from the combination. 23. Dubious discuss. The red phycoerythrin pigment is an adaptation to help red algae catch more sunlight in deep water. 23. As such, some red algae that live in shallow water have less phycoerythrin in their rhodoplasts, and can appear more greenish. 39. Rhodoplasts synthesize a form of starch called fluoridin starch. 23. Which collects into granules outside the rhodoplast, in the cytoplasm of the red alga. 12. Chloroplastida, green chloroplasts. See also, chloroplastida. The chloroplastida chloroplasts, or green chloroplasts, are another large, highly diverse primary chloroplast lineage and includes green algae and land plants. 40. They differ from glaucophyte and red algal chloroplasts in that they have lost their phycobilisms, and contain chlorophyll B instead. 12. Most green chloroplasts are green in color, though some aren't due to accessory pigments that override the chlorophyll's green colors, including some forms of Haematococcus pluvialis. Chloroplastida chloroplasts have lost the peptidoglycan wall between their double membrane, leaving an intermembrane space. 12. Some plants seem to have kept the genes for the synthesis of the peptidoglycan layer, though they've been repurposed for use in chloroplast division instead. 41. Green algae and plants keep their starch inside their chloroplasts. 12, 31, 40, and in plants and some algae, the chloroplast thylakoids are arranged in granar stacks. Some green algal chloroplasts contain a structure called a pyrenoid. 12, which is functionally similar to the glaucophyte carboxysm in that it is where rubisco and CO2 are concentrated in the chloroplast. 42. Helicosporidium is a genus of non-photosynthetic parasitic green algae that is thought to contain a vestigial chloroplast. 31. Genes from a chloroplast. 43. And nuclear genes indicating the presence of a chloroplast have been found in Helicosporidium. 31. Even if nobody's seen the chloroplast itself. 31. Paulinella chromatophora. See also, Paulinella. The photosynthetic amoeboids in the genus Paulinella, P. chromatophora, P. micropora, and marine P. longichromatophora, have the only known independently evolved chloroplast, often called a chromatophora. Note 1. While all other chloroplasts originate from a single ancient endosymbiotic event, Paulinella independently acquired an endosymbiotic cyanobacterium from the genus Synechococcus around 90 to 140 million years ago. 27, 29. Each Paulinella cell contains one or two sausage-shaped chloroplasts. 21, 44. They were first described in 1894 by German biologist Robert Lauterborn. 45. The chromatophore is highly reduced compared to its free-living cyanobacterial relatives and has limited functions. For example, it has a genome of about 1 million base pairs, 1/3 the size of Synechococcus genomes, and only encodes around 850 proteins. 21. However, explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Light micrograph of the amoeboid. Paulinella chromatophora. Euglena, a euglenophite. Contains secondary chloroplasts. From green algae. Chlorarachnion reptans is a chlorarachniophyte. Chlorarachniophytes replaced their original red algal endosymbiont with a green alga. This is still much larger than other chloroplast genomes, which are typically around 150,000 base pairs. Chromatophoras have also transferred much less of their DNA to the nucleus of their hosts. About 0.8% of the nuclear DNA in Paulinella is from the chromatophora, compared with 11–14% from the chloroplast in plants. 44. Similar to other chloroplasts, Paulinella provides specific proteins to the chromatophora using a specific targeting sequence. 46. Because chromatophoras are much younger compared to the canonical chloroplasts, Paulinella chromatophora is studied to understand how early chloroplasts evolved. 21. Secondary and tertiary chloroplast lineages. Green algal-derived secondary chloroplasts. Green algae have been taken up by the euglenids, chlorarachniophytes, a lineage of dinoflagellates. 31. And possibly the ancestor of the cache lineage, cryptomonids, alveolates, stramenophiles and haptophytes. 47. In 3 or 4 separate events. 48. Many green algal-derived chloroplasts contain pyrenoids, but unlike chloroplasts in their green algal ancestors, storage product collects in granules outside the chloroplast. 12. Euglenophytes. Euglenophytes are a group of common flagellated protists that contain chloroplasts derived from a green alga. 29. Euglenophyte chloroplasts have 3 membranes. It is thought that the membrane of the primary endosymbiont was lost, leaving the cyanobacterial membranes, and the secondary hosts. Phagosomal membrane. 29. Euglenophyte chloroplasts have a pyrenoid and thylakoids stacked in groups of 3. Photosynthetic product is stored in the form of paramylin, which is contained in membrane-bound granules in the cytoplasm of the euglenophyte. 12. 31. Chlorarachniophytes. Chlorarachniophytes /KLR-rachniophytes are a rare group of organisms that also contain chloroplasts derived from green algae. 29. Though their story is more complicated than that of the euglenophytes. The ancestor of chlorarachniophytes is thought to have been a eukaryote with a red algal-derived chloroplast. It is then thought to have lost its first red algal chloroplast, and later engulfed a green alga, giving it its second, green algal-derived chloroplast. 31. Chlorarachniophyte chloroplasts are bounded by 4 membranes, except near the cell membrane, where the chloroplast membranes fuse into a double membrane. 12. Their thylakoids are arranged in loose stacks of 3. 12. Chlorarachniophytes have a form of polysaccharide called chrysolaminorin, which they store in the cytoplasm. 31. Often collected around the chloroplast pyrenoid, which bulges into the cytoplasm. 12. Chlorarachniophyte chloroplasts are notable because the green alga they are derived from has not been completely broken down. Its Nucleus still persists as a nucleomorph. 29. Found between the second and third chloroplast membranes. 12. The periplastide space, which corresponds to the green alga's cytoplasm. 31. Prasinophyte-derived dinophyte chloroplast. Lepidodinium viride and its close relatives are dinophytes, see below, that lost their original peridinin chloroplast and replaced it with a green algal-derived chloroplast, more specifically, a prasinophyte. 1249. Lepidodinium is the only dinophyte that has a chloroplast that's not from the rhodoplast lineage. The chloroplast is surrounded by two membranes and has no nucleomorph. All the nucleomorph genes have been transferred to the dinophyte nucleus. 49. The endosymbiotic event that led to this chloroplast was serial secondary endosymbiosis. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Scanning electron micrograph of Gephora oceanica, a haptophyte. The photosynthetic pigments present in their chloroplasts make diatoms greenish-brown. Rather than tertiary endosymbiosis, the endosymbiont was a green alga containing a primary chloroplast, making a secondary chloroplast. 31. Red algal-derived chloroplasts. Cryptophytes. Cryptophytes, or cryptomonids, are a group of algae that contain a red algal-derived chloroplast. Cryptophyte chloroplasts contain a nucleomorph that superficially resembles that of the chlorarachniophytes. 29. Cryptophyte chloroplasts have four membranes, the outermost of which is continuous with the rough endoplasmic reticulum. They synthesize ordinary starch, which is stored in granules found in the periplastide space, outside the original double membrane, in the place that corresponds to the red alga's cytoplasm. Inside cryptophyte chloroplasts is a pyrenoid and thylakoids in stacks of 2. 12. Their chloroplasts do not have phocobilisms, 12, but they do have phocobilin pigments which they keep in their thylakoid space, rather than anchored on the outside of their thylakoid membranes. 1229. Cryptophytes may have played a key role in the spreading of red algal-based chloroplasts. 51. Haptophytes. Haptophytes are similar and closely related to cryptophytes or heterokontophytes. 31. Their chloroplasts lack a nucleomorph. 1229. Their thylakoids are in stacks of 3, and they synthesize chrysolaminoin sugar, which they store completely outside of the chloroplast, in the cytoplasm of the haptophyte. 12. Stramenophiles, heterokontophytes. The stramenophiles, also known as heterokontophytes, are a very large and diverse group of eukaryotes. It includes Acrophyta, which includes diatoms, brown algae, seaweeds, and golden algae, chrysophytes. 39. And Xanthophyceae, also called yellow-green algae. 31. Heterokont chloroplasts are very similar to haptophyte chloroplasts. They have a pyrenoid, triplet thylakoids, and, with some exceptions, 12.4. Layoplastidic envelope, the outermost epiplastide membrane connected to the endoplasmic reticulum. Like haptophytes, stramenophiles store sugar in chrysolaminoin granules in the cytoplasm. 12. Stramenophile. Chloroplasts contain chlorophyll and, with a few exceptions, 12. Chlorophyll C, 29, but also have carotenoids, which give them their many colors. 39. apicomplexans, chromarids, and dinophytes. The alveolates are a major clade of unicellular eukaryotes of both autotrophic and heterotrophic members. The most notable shed characteristic is the presence of cortical, outer region, alveoli, sacs. These are flattened vesicles, sacs, packed into a continuous layer just under the membrane and supporting it, typically forming a flexible pellicle, thin skin. In dinoflagellates they often form armor plates. Many members contain a red algal-derived plastide. One notable characteristic of this diverse group is the frequent loss of photosynthesis. However, a majority of these heterotrophs continue to process a non-photosynthetic plastide. 52. Apicomplexans. Apicomplexans are a group of alveolates. Like the helicosporidea, they are parasitic, and have a non-photosynthetic chloroplast. 31. They were once thought to be related to the helicosporidea, but it is now known that the helicosporidea are green algae rather than part of the cash lineage. 31. The apicomplexans include Plasmodium, the malaria parasite. Many apicomplexans keep a vestigial red algal-derived chloroplast, 53, 31, called an apicoplast, which they inherited from their ancestors. Other apicomplexans like Cryptosporidium have lost the chloroplast completely. 53. Apicomplexans store their energy in amylopectin granules that are located in their cytoplasm, even though they are non-photosynthetic. 12. Apicoplasts have lost all photosynthetic function, and contain no photosynthetic pigments or true thylakoids. They are bounded by 4 membranes, but the membranes are not connected to the endoplasmic reticulum. 12. The fact that apicomplexans still keep their non-photosynthetic chloroplast around demonstrates how the chloroplast carries out important functions other than photosynthesis. Plant. Chloroplasts provide plant cells with many important things besides sugar, and apicoplasts are no different. They synthesize fatty acids, isopentenyl pyrophosphate, iron-sulfur clusters, and carry out part of the HEM pathway. 53. This makes the apicoplast an attractive target. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Keratium furca, a peridinin containing dinophyte, 56. Carinea brevis is a fucoxanthin containing dinophyte responsible for algal blooms called red tides. 49. 4 drugs to cure apicomplexan-related diseases. 23. The most important apicoplast function is isopentenyl pyrophosphate synthesis. In fact, apicomplexans die when something interferes with this apicoplast function, and when apicomplexans are grown in an isopentenyl pyrophosphate-rich medium, they dump the organelle. 53. Chromarids. The Chromrida is a newly discovered group of algae from Australian corals which comprises some close photosynthetic relatives of the apicomplexans. The first member, Chromarivalia, was discovered and first isolated in 2001. The discovery of Chromarivalia with similar structure to the apicomplexans provides an important link in the evolutionary history of the apicomplexans and dinophytes. Their plastids have 4 membranes, lack chlorophyll C and use the type II form of rubisco obtained from a horizontal transfer event. 54. Dinophytes. The dinoflagellates are yet another very large and diverse group of protists, around half of which are, at least partially, photosynthetic. 39, 49. Most dinophyte chloroplasts are secondary red algal-derived chloroplasts. Many other dinophytes have lost the chloroplast, becoming the non-photosynthetic kind of dinoflagellate, or replaced it through tertiary endosymbiosis. 55. The engulfment of another eukaryotic algae containing a red algal-derived chloroplast. Others replaced their original chloroplast with a green algal-derived one. 29, 31, 49. Most dinophyte chloroplasts contain form 2 rubisco, at least the photosynthetic pigments chlorophyll A, chlorophyll C2, beta-carotene, and at least one dinophyte unique xanthophyll, peridinin, dinocanthin, or diadenocanthin, giving many a golden brown color. 52, 49. All dinophytes store starch in their cytoplasm, and most have chloroplasts with thylakoids arranged in stacks of 3. 12. The most common dinophyte chloroplast is the peridinin-type chloroplast, characterized by the carotenoid pigment peridinin in their chloroplasts, along with chlorophyll A and chlorophyll C2. 29, 49. Peridinin is not found in any other group of chloroplasts. 49. The peridinin chloroplast is bounded by 3 membranes, occasionally 2. 12. Having lost the red algal endosymbiont's original cell membrane. 29, 31. The outermost membrane is not connected to the endoplasmic reticulum. 12, 49. They contain a pyrenoid, and have triplet-stacked thylakoids. Starch is found outside the chloroplast. 12. An important feature of these chloroplasts is that their chloroplast DNA is highly reduced and fragmented into many small circles. Most of the genome has migrated to the nucleus, and only critical photosynthesis-related genes remain in the chloroplast. 49. The peridinin chloroplast is thought to be the dinophyte's original chloroplast. 49, which has been lost, reduced, replaced, or has company in several other dinophyte lineages. 31. Tertiary chloroplasts, haptophyte-derived. The few coxanthin dinophyte lineages, including Carlodinium and Carinea, 31, lost their original red algal-derived chloroplast, and replaced it with a new chloroplast derived from a haptophyte endosymbiont, making these tertiary plastids. Carlodinium and Carinea probably took up different heterocontophytes. 31. Because the haptophyte chloroplast has 4 membranes, tertiary endosymbiosis would be expected to create a 6-membraned chloroplast, adding the haptophyte cell membrane and the dinophyte's phagosomal vacuole. 57. However, the haptophyte was heavily reduced, stripped of a few membranes and its nucleus, leaving only its chloroplast, with its original double membrane, and possibly 1 or 2 additional membranes around it. 31, 57. Fucoxanthin-containing chloroplasts are characterized by having the pigment fucoxanthin, actually 19 hexanoyloxy fucoxanthin and / or 19 butanoyloxy fucoxanthin, and no peridinin. Fucoxanthin is also found in haptophyte chloroplasts, providing evidence of ancestry. 49. Diatom-derived dinophyte chloroplasts. Some dinophytes, like Cryptoperidinium and Durinschia, 31, have a diatom, heterocontophyte, derived chloroplast. 29. These chloroplasts are bounded by up to 5 membranes, 29, depending on whether the entire diatom endosymbiont is counted as the chloroplast, or just the red algal-derived chloroplast inside it. The diatom endosymbiont has been reduced relatively little. It still retains its original mitochondria, 31, and has endoplasmic reticulum, ribosomes, a nucleus, and of course, red algal-derived chloroplasts, practically a complete cell, 58, all inside the host's endoplasmic reticulum lumen. 31. However, the diatom endosymbiont can't explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Dinophysis acuminator has chloroplasts taken from a cryptophyte. 29. Store its own food. Its storage polysaccharide is found in granules in the dinophyte host's cytoplasm. Instead. 12. 58. The diatom endosymbiont's nucleus is present, but it probably can't be called a nucleomorph, because it shows no sign of genome reduction, and might have even been expanded. 31. Diatoms have been engulfed by dinoflagellates at least 3 times. 31. The diatom endosymbiont is bounded by a single membrane. 49. Inside it are chloroplasts with 4 membranes. Like the diatom endosymbiont's diatom ancestor, the chloroplasts have triplet thylakoids and pyrenoids. 58. In some of these genera, the diatom endosymbiont's chloroplasts aren't the only chloroplasts in the dinophyte. The original 3 membraned peridinin chloroplast is still around, converted to an ispot. 29, 31. Kleptoplasty. Main article, Kleptoplasty. In some groups of mixotrophic protists, like some dinoflagellates, for example dinophyses, chloroplasts are separated from a captured alga and used temporarily. These kleptochloroplasts may only have a lifetime of a few days and are then replaced. 59, 60. Cryptophyte-derived dinophyte chloroplast. Members of the genus Dinophyses have a phacobilin-containing, 57, chloroplast taken from a cryptophyte. 29. However, the cryptophyte is not an endosymbiont, only the chloroplast seems to have been taken, and the chloroplast has been stripped of its nucleomorph and outermost 2 membranes, leaving just a 2-membraned chloroplast. Cryptophyte chloroplasts require their nucleomorph to maintain themselves. And Dinophysis species grown in cell culture alone cannot survive, so it is possible, but not confirmed, that the Dinophysis chloroplast is a kleptoplast. If so, Dinophysis chloroplasts wear out and Dinophysis species must continually engulf cryptophytes to obtain new chloroplasts to replace the old ones. 49. Chloroplast DNA. Main article, Chloroplast DNA. See also, list of sequenced plastoms. Chloroplasts, like other endosymbiotic organelles, contain a genome separate from that in the cell nucleus. The existence of chloroplast DNA, cpDNA, was identified biochemically in 1959, 61, and confirmed by electron microscopy in 1962. 62. The discoveries that the chloroplast contains ribosomes, 63, and performs protein synthesis, 64, revealed that the chloroplast is genetically semi-autonomous. Chloroplast DNA was first sequenced in 1986. 65. Since then, hundreds of chloroplast genomes from various species have been sequenced, but they are mostly those of land plants and green algae. Glaucophytes, red algae, and other algal groups are extremely underrepresented, potentially introducing some bias in views of typical chloroplast DNA structure and content. 66. Molecular structure. With few exceptions, most chloroplasts have their entire chloroplast genome combined into a single large circular DNA molecule. 66. Typically 120,000,170,000 base pairs long. 67, 68, 69, 18, and a mass of about 81–30 million Daltons. 70. While chloroplast genomes can almost always be assembled into a circular map, the physical DNA molecules inside cells take on a variety of linear and branching forms. 66, 71. New chloroplasts may contain up to 100 copies of their genome. 67, though the number of copies decreases to about 15–20 as the chloroplasts age. 72. Chloroplast DNA is usually condensed into nucleids, which can contain multiple copies of the chloroplast genome. Many nucleids can be found in each chloroplast. 70. In primitive red algae, the chloroplast DNA nucleids are clustered in the center of the chloroplast, while in green plants and green algae, the nucleids are dispersed throughout the stroma. 73. Chloroplast DNA is not. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Chloroplast DNA replication via multiple D-loop mechanisms. Adapted from Krishnan N.M., Rao B.J.'s paper, A Comparative Approach to Elucidate Chloroplast Genome Replication. Over time, base changes in the DNA sequence can arise from deamination mutations. When adenine is deaminated, it becomes hypoxanthine, which can pair with cytosine. During replication, the cytosine will pair with guanine, causing an A-G base change. Associated with true histones, proteins that are used to pack DNA molecules tightly in eukaryote nuclei. 17. Though in red algae, similar proteins tightly pack each chloroplast DNA ring in a nucleid. 73. Many chloroplast genomes contain two inverted repeats, which separate a long single copy section, LSC, from a short single copy section, SSC. 69. A given pair of inverted repeats are rarely identical, but they are always very similar to each other, apparently resulting from concerted evolution. 66. The inverted repeats vary wildly in length, ranging from 4,000 to 25,000 base pairs long each and containing as few as 4 or as many as over 150 genes. 66. The inverted repeat regions are highly conserved in land plants, and accumulate few mutations. 69, 74. Similar inverted repeats exist in the genomes of cyanobacteria and the other two chloroplast lineages, Glaucophyta and Rhodophyceae, suggesting that they predate the chloroplast. 66. Some chloroplast genomes have since lost, 74, 75, or flipped the inverted repeats, making them direct repeats. 66. It is possible that the inverted repeats help stabilize the rest of the chloroplast genome, as chloroplast genomes which have lost some of the inverted repeat segments tend to get rearranged more. 75. DNA repair and replication. In chloroplasts of the Mosfisca patterns, the DNA mismatch repair protein MSH1 interacts with the recombinational repair proteins Rec A and Rec G to maintain chloroplast genome stability. 76. In chloroplasts of the plant Arabidopsis thaliana, the Rec A protein maintains the integrity of the chloroplast's DNA by a process that likely involves the recombinational repair of DNA damage. 77. The mechanism for chloroplast DNA, cpDNA, replication has not been conclusively determined, but two main models have been proposed. Scientists have attempted to observe chloroplast replication via electron microscopy since the 1970s. 78, 79. The results of the microscopy experiments led to the idea that chloroplast DNA replicates using a double displacement loop, D-loop. As the D-loop moves through the circular DNA, it adopts a theta intermediary form, also known as a Cairns replication intermediate, and completes replication with a rolling circle mechanism. 78, 80. Transcription starts at specific points of origin. Multiple replication. Forks open up, allowing replication machinery to transcribe the DNA. As replication continues, the forks grow and eventually converge. The new cpDNA structures separate, creating daughter cpDNA chromosomes. In addition to the early microscopy experiments, this model is also supported by the amounts of deamination seen in cpDNA. 78. Deamination occurs when an amino group is lost and is a mutation that often results in base changes. When adenine is deaminated, it becomes hypoxanthine. Hypoxanthine can bind to cytosine, and when the XC base pair is replicated, it becomes AGC, thus, an AG base change. 81. In cpDNA, there are several AG deamination gradients. DNA becomes susceptible to deamination events when it is single-stranded. When replication forks form, the strand not being copied is single-stranded, and thus at risk for AG deamination. Therefore, gradients in deamination indicate that replication forks were most likely present and the direction that they initially opened. The highest gradient is most likely nearest the start site because it was single-stranded for the longest amount of time. 78. This mechanism is still the leading theory. Today, however, a second theory suggests that most cpDNA is actually linear and replicates through homologous recombination. It further contends that only a minority of the genetic material is kept in circular chromosomes while the rest is in branched, linear, or other complex structures. 78, 80. One of competing model for cpDNA replication asserts that most cpDNA is linear and participates in homologous recombination and replication structures similar to the linear and circular DNA structures of bacteriophage T4. 80, 82. It has been established that some plants have linear cpDNA, such as maize, and that more species still contain complex structures that scientists do not yet understand. 80. When the original experiments on cpDNA were performed, scientists did notice linear structures. However, they attributed these linear forms to broken circles. 80. If the branched and complex structures seen in cpDNA experiments are real and not artifacts of concatenated circular cytochrome photosystem I, acetylcocarboxylase, rubisco, tinase, tRNA, photosystem II, tinase, tRNA, photo, Ribo. Pro. TR. tRNA. NADDO. Ribosoma. tRNA. Replication origin regions tRNA. Small RNA. Ribosomal protein. Replication origin regions. Ribosomal RNA. TNAs. Ribosomal RNA. tRNA. Cytochromes. Photosystem II. Ribosomal proteins. Photosystem I. Cytochromes. Photosystem II. ATP synthase. TNAs. NAD dehydrogenase. tRNA ribosomal proteins. Photosystem I. TNAs. Photosystem II. RNA polymerase. Ribosomal protein. ATP synthase. TNAs. Ribosomal protein. tRNA. Photosystem II. tRNA. tRNA. Ribosomal RNA. tRNA. Ribosomal RNA. tRNA ribosomal protein. Photosystem I. NAD dehydrogenase. tRNA. Ribosomal protein. NAD dehydrogenase. tRNA. tRNA. Ribosomal proteins. Initiation factor 1. Ribosomal proteins. RNA polymerase. ATP-dependent protease. Ribosomal proteins. TNAs. Edit times IM. Chloroplast DNA interactive gene map of chloroplast DNA from Nicotiana tabacum. Segments with labels on the inside are on the B strand of DNA, segments with labels on the outside are on the A strand. Notches indicate introns. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. DNA or broken circles, then a D-loop mechanism of replication is insufficient to explain how those structures would replicate. 80. At the same time, homologous recombination does not expand the multiple A-G gradients seen in plastoms. 78. Because of the failure to explain the deamination gradient as well as the numerous plant species that have been shown to have circular cpDNA, the predominant theory continues to hold that most cpDNA is circular and most likely replicates via a D-loop mechanism. Gene content and protein synthesis. The ancestral cyanobacteria that led to chloroplasts probably had a genome that contained over 3,000 genes, but only approximately 100 genes remain in contemporary chloroplast genomes. 18, 22, 68. These genes code for a variety of things, mostly to do with the protein pipeline and photosynthesis. As in prokaryotes, genes in chloroplast DNA are organized into operons. 22. Unlike prokaryotic DNA molecules, chloroplast DNA molecules contain introns. Plant mitochondrial DNAs do too, but not human mTNAs. 83. Among land plants, the contents of the chloroplast genome are fairly similar. 69. Chloroplast genome reduction and gene transfer. Over time, many parts of the chloroplast genome were transferred to the nuclear genome of the host. 67, 68, 84, a process called endosymbiotic gene transfer. As a result, the chloroplast genome is heavily reduced compared to that of free-living cyanobacteria. Chloroplasts may contain 6,100 genes whereas cyanobacteria often have more than 1,500 genes in their genome. 85. Recently, a plastide without a genome was found, demonstrating chloroplasts can lose their genome during endosymbiotic the gene transfer process. 86. Endosymbiotic gene transfer is how we know about the lost chloroplasts in many cache lineages. Even if a chloroplast is eventually lost, the genes it donated to the former host's nucleus persist, providing evidence for the lost chloroplast's existence. For example, while diatoms, a heterokontophyte, now have a red algal-derived chloroplast, the presence of many green algal genes in the diatom nucleus provide evidence that the diatom ancestor had a green algal-derived chloroplast at some point, which was subsequently replaced by the red chloroplast. 47. In land plants, some 11–14% of the DNA in their nuclei can be traced back to the chloroplast, 44, up to 18% in Arabidopsis, corresponding to about 4,500 protein-coding genes. 87. There have been a few recent transfers of genes from the chloroplast DNA to the nuclear genome in land plants. 68. Of the approximately 3,000 proteins found in chloroplasts, some 95% of them are encoded by nuclear genes. Many of the chloroplasts' protein complexes consist of subunits from both the chloroplast genome and the host's nuclear genome. As a result, protein synthesis must be coordinated between the chloroplast and the nucleus. The chloroplast is mostly under nuclear control, though chloroplasts can also give out signals regulating gene expression in the nucleus, called retrograde signaling. 88. Recent research indicates that parts of the retrograde signaling network once considered characteristic for land plants emerged already in an algal progenitor. 89, 90, 91, integrating into expressed cohorts of genes in the closest algal relatives of land plants. 92. Protein synthesis. See also, transcription and translation. Protein synthesis within chloroplasts relies on two RNA polymerases. One is coded by the chloroplast DNA, the other is of nuclear origin. The two RNA polymerases may recognize and bind to different kinds of promoters within the chloroplast genome. 93. The ribosomes in chloroplasts are similar to bacterial ribosomes. 94. This section needs expansion with genome size differences between algae and land plants. Chloroplast stuff coded by the nucleus. You can help by adding to it. January 2013. Protein targeting and import. See also, translation. Because so many chloroplast genes have been moved to the nucleus, many proteins that would originally have been translated in the chloroplast are now synthesized in the cytoplasm of the plant cell. These proteins must be directed back to the chloroplast, and imported through at least two chloroplast membranes. 95. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The two ends of a polypeptide are called the N terminus, or amino end, and the C terminus, or carboxyl end. 96. This polypeptide has 4 amino acids linked together. At the left is the N terminus, with its amino, H2N, group in green. The blue C terminus, with its carboxyl group, CO2H, is at the right. Transmission electron. Microscope image of a chloroplast. Grana of the lacoids. And their connecting lamellae are clearly visible. Curiously, around half of the protein products of transferred genes aren't even targeted back to the chloroplast. Many became exaptations, taking on new functions like participating in cell division, protein routing, and even disease resistance. A few chloroplast genes found new homes in the mitochondrial genome. Most became non-functional pseudogenes, though a few tRNA genes still work in the mitochondrion. 85. Some transferred chloroplast DNA protein products get directed to the secretory pathway. 85. Though many secondary plastids are bounded by an outermost membrane derived from the host's cell membrane, and therefore topologically outside of the cell. Because to reach the chloroplast from the cytosol, the cell membrane must be crossed, which signifies entrance into the extracellular space. In those cases, chloroplast-targeted proteins do initially travel along the secretory pathway. 31. Because the cell acquiring a chloroplast already had mitochondria, and peroxisms, and a cell membrane for secretion, the new chloroplast host had to develop a unique protein targeting system to avoid having chloroplast proteins being sent to the wrong organelle. 95. In most, but not all cases, nuclear-encoded chloroplast proteins are translated with a cleavable transit peptide that's added to the N-terminus of the protein precursor. Sometimes the transit sequence is found on the C-terminus of the protein. 97, or within the functional part of the protein. 95. Transport proteins and membrane translocans. After a chloroplast polypeptide is synthesized on a ribosome in the cytosol, an enzyme specific to chloroplast proteins, 98, phosphorylates, or adds a phosphate group to many, but not all, of them in their transit sequences. 95. Phosphorylation helps many proteins bind the polypeptide, keeping it from folding prematurely. 95. This is important because it prevents chloroplast proteins from assuming their active form and carrying out their chloroplast functions in the wrong place, the cytosol. 99. 100. At the same time, they have to keep just enough shape so that they can be recognized by the chloroplast. 99. These proteins also help the polypeptide get imported into the chloroplast. 95. From here, chloroplast proteins bound for the stroma must pass through two protein complexes, the TOC complex, or T-translocan on the OU to chloroplast membrane, and the TIC translocan, or T-translocan on the inochloroplast membrane translocan. 95. Chloroplast polypeptide chains probably often travel through the two complexes at the same time, but the TIC complex can also retrieve preproteins lost in the intermembrane space. 95. Structure. In land plants, chloroplasts are generally lens-shaped, 310 m in diameter and 13 m thick. 101, 18. Corn seedling chloroplasts are almost equal to 20 m3 in volume. 18. Greater diversity in chloroplast shapes exists among the algae, which often contain a single chloroplast. 12. That can be shaped like a net, for example, Edogonium, 102, a cup, for example, Chlamydomonas, 103, a ribbon-like spiral around the edges of the cell. For example, Spirogyra, 104, or slightly twisted bands at the cell edges, for example, Cyrogonium. 105. Some algae have two chloroplasts in each cell. They are star-shaped in Zinuma, 106, or may follow the shape of half the cell in order desmidials. 107. In some algae, the chloroplast takes up most of the cell, with pockets for the nucleus and other organelles. 12. For example, some species of chlorella have a cup-shaped chloroplast that occupies much of the cell. 108. All chloroplasts have at least three membrane systems: the outer chloroplast membrane, the inner chloroplast membrane, and the thylakoid system. The two innermost lipid bilayer membranes, 109, that surround all chloroplasts correspond to the outer and inner membranes of the ancestral cyanobacterium's gram-negative cell wall, 29, 110, 111, and not the phagosomal membrane from the host, which was probably lost. 29. Chloroplasts that are the product of secondary endosymbiosis may have additional membranes surrounding these three. 32. Inside the outer and inner chloroplast membranes is the chloroplast stroma, a semigel-like fluid. 23. That makes up much of a chloroplast's volume, and in which the thylakoid system floats. See also, chloroplast membrane. There are some common misconceptions about the outer and inner chloroplast membranes. The fact that chloroplasts are surrounded by a double membrane is often cited as evidence that they are the descendants of endosymbiotic cyanobacteria. This is often interpreted as meaning the outer chloroplast membrane is. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Chloroplast ultrastructure, interactive diagram. Chloroplasts have at least three distinct membrane systems, and a variety of things can be found in their stroma. Instead of an intermembrane space, glaucophyte algae have a peptidoglycan wall between their inner and outer chloroplast membranes. The product of the host cell membrane infolding to form a vesicle to surround the ancestral cyanobacterium, which is not true. Both chloroplast membranes are homologous to the cyanobacterium's original double membranes. 29. The chloroplast double membrane is also often compared to the mitochondrial double membrane. This is not a valid comparison. The inner mitochondria membrane is used to run proton pumps and carry out oxidative phosphorylation across to generate ATP energy. The only chloroplast structure that can considered analogous to it is the internal thylakoid system. Even so, in terms of in-out, the direction of chloroplast H and ion flow is in the opposite direction compared to oxidative phosphorylation in mitochondria. 23, 112. In addition, in terms of function, the inner chloroplast membrane, which regulates metabolite passage and synthesizes some materials, has no counterpart in the mitochondrion. 23. Outer chloroplast membrane. Main article, chloroplast membrane. The outer chloroplast membrane is a semiporous membrane that small molecules and ions can easily diffuse across. 113. However, it is not permeable to larger proteins, so chloroplast polypeptides being synthesized in the cell cytoplasm must be transported across the outer chloroplast membrane by the TOC complex, or T-translocon on the OU to C chloroplast membrane. 95. The chloroplast membranes sometimes protrude out into the cytoplasm, forming a stromule, or stroma containing tubule. Stromules are very rare in chloroplasts, and are much more common in other plastids like chromoplasts and amyloplasts in petals and roots, respectively. 114, 115. They may exist to increase the chloroplast's surface area for cross-membrane transport, because they are often branched and tangled with the endoplasmic reticulum. 116. When they were first observed in 1962, some plant biologists dismissed the structures as artifactual, claiming that stromules were just oddly shaped chloroplasts with constricted regions or dividing chloroplasts. 117. However, there is a growing body of evidence that stromules are functional, integral features of plant cell plastids, not merely artifacts. 118. Intermembrane space and peptidoglycan wall. Usually, a thin intermembrane space about 10-20 nanometers thick exists between the outer and inner chloroplast membranes. 119. Glaucophyte algal chloroplasts have a peptidoglycan layer between the chloroplast membranes. It corresponds to the peptidoglycan cell wall of their cyanobacterial ancestors, which is located between their two cell membranes. These chloroplasts are called muroplasts, from Latin mura, meaning wall. Other chloroplasts were assumed to have lost the cyanobacterial wall, leaving an intermembrane space between the two chloroplast envelope membranes, 23, but has since been found also in moss, lycophytes and ferns. 120. Inner chloroplast membrane. Main article, chloroplast membrane. The inner chloroplast membrane borders the stroma and regulates passage of materials in and out of the chloroplast. After passing through the TOC complex in the outer chloroplast membrane, polypeptids must pass through the TIC complex, T-translocon on the inner chloroplast membrane, which is located in the inner chloroplast membrane. 95. In addition to regulating the passage of materials, the inner chloroplast membrane is where fatty acids, lipids, and carotenoids are synthesized. 23. Peripheral reticulum. Some chloroplasts contain a structure called the chloroplast peripheral reticulum. 119. It is often found in the chloroplasts of C4 plants, though it has also been found in some C3 angiosperms, 23, and even some gymnosperms. 121. The chloroplast peripheral reticulum consists. Edit time source image. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Chloroplast ribosomes comparison of a chloroplast ribosome, green, and a bacterial ribosome, yellow. Important features common to both ribosomes and chloroplast. Unique features are labeled of a maze of membranous tubes and vesicles continuous with the inner chloroplast membrane that extends into the internal stromal fluid of the chloroplast. Its purpose is thought to be to increase the chloroplast's surface area for cross-membrane transport between its stroma and the cell cytoplasm. The small vesicles sometimes observed may serve as transport vesicles to shuttle stuff between the thylakoids and intermembrane space. 122. Stroma. Main article, stroma. The protein-rich, 23, alkaline, 112, aqueous fluid within the inner chloroplast membrane and outside of the thylakoid space is called the stroma, 23, which corresponds to the cytosol of the original cyanobacterium. Nucleids of chloroplast DNA, chloroplast ribosomes, the thylakoid system with plastoglobuli, starch granules, and many proteins can be found floating around in it. The Calvin cycle, which fixes CO2 into G3P takes place in the stroma. Chloroplast ribosomes. Chloroplasts have their own ribosomes, which they use to synthesize a small fraction of their proteins. Chloroplast ribosomes are about 2/3 the size of cytoplasmic ribosomes. Around 17 nm vs 25 nm. 119. They take mRNAs transcribed from the chloroplast DNA and translate them into protein. While similar to bacterial ribosomes, 17, chloroplast translation is more complex than in bacteria, so chloroplast ribosomes include some chloroplast unique features. 123, 124. Small subunit ribosomal RNAs in several chlorophyta and euglonid chloroplasts lack motifs for Shine-Dalgarno sequence recognition. 125, which is considered essential for translation initiation in most chloroplasts and prokaryotes. 126, 127, such loss is also rarely observed in other plastids and prokaryotes. 125, 128, an additional 4.5s RNA with homology to the 3' of 23s is found in higher plants. 124. Plastoglobuli. Plastoglobuli, singular plastoglobulus, sometimes spelled plastoglobule, s, are spherical bubbles of lipids and proteins, 23, about 45-60 nanometers across. 129, they are surrounded by a lipid monolayer. 129, plastoglobuli are found in all chloroplasts. 119, but become more common when the chloroplast is under oxidative stress. 129, or when it ages and transitions into a gerontoplast. 23. Plastoglobuli also exhibit a greater size variation under these conditions. 129. They are also common in etioplasts, but decrease in number as the etioplasts mature into chloroplasts. 129. Plastoglobuli contain both structural proteins and enzymes involved in lipid synthesis and metabolism. They contain many types of lipids, including plastoquinone, vitamin E, carotenoids and chlorophylls. 129. Plastoglobuli were once thought to be free-floating in the stroma, but it is now thought that they are permanently attached either to a thylakoid or to another plastoglobulus attached to a thylakoid, a configuration that allows a plastoglobulus to exchange its contents with the thylakoid network. 129. In normal green chloroplasts, the vast majority of plastoglobuli occur singularly, attached directly to their parent thylakoid. In old or stressed chloroplasts, plastoglobuli tend to occur in linked groups or chains, still always anchored to a thylakoid. 129. Plastoglobuli form when a bubble appears between the layers of the lipid bilayer of the thylakoid membrane, or bud from existing plastoglobuli, though they never detach and float off into the stroma. 129. Practically all plastoglobuli form on or near the highly curved edges of the thylakoid discs or sheets. They are also more common on stromal thylakoids than on granule ones. 129. Starch granules. Starch granules are very common in chloroplasts, typically taking up 15% of the organelle's volume. 130. Though in some other plastids like amyloplasts, they can be big enough to distort the shape of the organelle. 119. Starch granules are simply accumulations of starch in the stroma, and are not bounded by a membrane. 119. Starch granules appear and grow throughout the day, as the chloroplast synthesizes sugars, and are consumed at night to fuel respiration and continue sugar export into the phloem. 131. Though in mature chloroplasts, it is rare for a starch granule to be completely consumed or for a new granule to accumulate. 130. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Transmission electron micrograph of Chlamydomonas reinhardtii, a green alga that contains a pyrenoid surrounded by starch. Rubisco, shown here in a space-filling model, is the main enzyme responsible for carbon fixation in chloroplasts. Starch granules vary in composition and location across different chloroplast lineages. In red algae, starch granules are found in the cytoplasm rather than in the chloroplast. 132. In C4 plants, mesophyll chloroplasts, which do not synthesize sugars, lack starch granules. 23. Rubisco. Main article, Rubisco. The chloroplast stroma contains many proteins, though the most common and important is Rubisco, which is probably also the most abundant protein on the planet. 112. Rubisco is the enzyme that fixes CO2 into sugar molecules. In C3 plants, Rubisco is abundant in all chloroplasts, though in C4 plants, it is confined to the bundle sheath chloroplasts, where the Calvin cycle is carried out in C4 plants. 133. Pyrenoids. Main article, pyrenoid. The chloroplasts of some hornworts, 134, and algae contain structures called pyrenoids. They are not found in higher plants. 135. Pyrenoids are roughly spherical and highly refractive bodies which are a site of starch accumulation in plants that contain them. They consist of a matrix opaque to electrons, surrounded by two hemispherical starch plates. The starch is accumulated as the pyrenoids mature. 136. In algae with carbon-concentrating mechanisms, the enzyme Rubisco is found in the pyrenoids. Starch can also accumulate around the pyrenoids when CO2 is scarce. 135. Pyrenoids can divide to form new pyrenoids, or be produced de novo. 136, 137. Thylakoid system. Main article, thylakoid. Thylakoids, sometimes spelled thylakoids, 139, are small interconnected sacs which contain the membranes that the light reactions of photosynthesis take place on. The word thylakoid comes from the Greek word thylakos, which means sac. 140. Suspended within the chloroplast stroma is the thylakoid system, a highly dynamic collection of membranous sacs called thylakoids where chlorophyll is found and the light reactions of photosynthesis happen. 11. In most vascular plant chloroplasts, the thylakoids are arranged in stacks called grana. 141, though in certain C4 plant chloroplasts, 133, and some algal chloroplasts, the thylakoids are free-floating. 12. Thylakoid structure. Using a light microscope, it is just barely possible to see tiny green granules, which were named grana. 119. With electron microscopy, it became possible to see the thylakoid system in more detail, revealing it to consist of stacks of flat thylakoids which made up the grana, and long interconnecting stromal thylakoids which linked different grana. 119. In the transmission electron microscope, thylakoid membranes appear as alternating light and dark bands, 8.5 nanometers thick. 119. The three-dimensional structure of the thylakoid membrane system has been disputed. Many models have been proposed, the most prevalent being the helical model, in which granum stacks of thylakoids are wrapped by helical stromal thylakoids. 142. Another model, known as the bifurcation model, which was based on the first electron tomography study of plant thylakoid membranes, depicts the stromal membranes as wide lamellar sheets perpendicular to the grana columns which bifurcates into multiple parallel discs forming the granum stroma assembly. 143. The helical model was supported by several additional works, 141, 144, but ultimately it was determined in 2019 that features from both the helical and bifurcation models are consolidated by newly discovered left-handed helical membrane junctions. 138. Likely for ease, the thylakoid system is still commonly depicted by older hub-and-spoke models where the grana are connected to each other by tubes of stromal thylakoids. 145. Grana consist of stacks of flattened circular granule thylakoids that resemble pancakes. Each granum can contain anywhere from 2 to 100 thylakoids. 119, though grana with 10-20 thylakoids are most common. 141. Wrapped around the grana are multiple parallel right-handed helical stromal thylakoids, also known as frets or lamellar thylakoids. The helices ascend at an angle of -20 degree, connecting to each granule thylakoid at a bridge-like slit junction. 141, 144, 138. The stroma lamellae extend as large sheets perpendicular to the grana columns. These sheets are connected to the right-handed helices, either directly or through bifurcations that form left-handed helical membrane surfaces. 138. The left-handed helical surfaces have a similar. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Scanning transmission electron microscope imaging of a chloroplast. Top 10 nm thick stem tomographic slice of a lettuce chloroplast. Grana stacks are interconnected by unstacked stromal thylakoids, called stroma lamellae. Round inclusions associated with the thylakoids are plastoglobules. Scale bar equals 200 nm. C. 138. Bottom, large-scale 3D model generated from segmentation of tomographic reconstructions by stem. Grana equals yellow, stroma lamellae equals green, plastoglobules equals purple, chloroplast envelope equals blue. C. 138. Granum stroma assembly structure. The prevailing model of the granum stroma assembly is stacks of granule thylakoids wrapped by right-handed helical stromal thylakoids which are connected to large parallel sheets of stromal thylakoids and adjacent right-handed helices by left-handed helical structures. Based on 138. Tilt angle to the right-handed helices. -20 degree, but a quarter the pitch. Approximately 4 left-handed helical junctions are present per granum, resulting in a pitch-balanced array of right and left-handed helical membrane surfaces of different radii and pitch that consolidate the network with minimal surface and bending. Energies. 138. While different parts of the thylakoid system contain different membrane proteins, the thylakoid membranes are continuous and the thylakoid space they enclose form a single continuous labyrinth. 141. Thylakoid composition. Embedded in the thylakoid membranes are important protein complexes which carry out the light reactions of photosynthesis. Photosystem II and photosystem I contain light harvesting complexes with chlorophyll and carotenoids that absorb light energy and use it to energize electrons. Molecules in the thylakoid membrane use the energized electrons to pump hydrogen ions into the thylakoid space, decreasing the pH and turning it acidic. ATP synthase is a large protein complex that harnesses the concentration gradient of the hydrogen ions in the thylakoid space to generate ATP energy as the hydrogen ions flow back out into the stroma, much like a dam turbine. 112. There are two types of thylakoids, granule thylakoids, which are arranged in grana, and stromal thylakoids, which are in contact with the stroma. Granule thylakoids are pancake-shaped circular discs about 300–600 nanometers in diameter. Stromal thylakoids are helicoid sheets that spiral around grana. 141. The flat tops and bottoms of granule thylakoids contain only the relatively flat photosystem II protein complex. This allows them to stack tightly, forming grana with many layers of tightly oppressed membrane, called granule membrane, increasing stability and surface area for light capture. 141. In contrast, photosystem I and ATP synthase are large protein complexes which jut out into the stroma. They can't fit in the oppressed granule membranes, and so are found in the stromal thylakoid membrane, the edges of the granule thylakoid discs and the stromal thylakoids. These large protein complexes may act as spacers between the sheets of stromal thylakoids. 141. The number of thylakoids and the total thylakoid area of a chloroplast is influenced by light exposure. Shaded chloroplasts contain larger and more grana with more thylakoid membrane area than chloroplasts exposed to bright light, which have smaller and fewer grana and less thylakoid area. Thylakoid extent can change within minutes of light exposure or removal. 122. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Deleseria sanguinea, a red alga, has chloroplasts that contain red pigments like phocoerythrin that mask their blue-green chlorophyll. A. 39. Pigments and chloroplast colors. Inside the photosystems embedded in chloroplast thylakoid membranes are various photosynthetic pigments, which absorb and transfer light energy. The types of pigments found are different in various groups of chloroplasts, and are responsible for a wide variety of chloroplast colorations. Other plastide types, such as the leucoplast and the chromoplast, contain little chlorophyll and do not carry out photosynthesis. Chlorophylls. Chlorophyll is found in all chloroplasts, as well as their cyanobacterial ancestors. Chlorophyll is a blue-green pigment. 146. partially responsible for giving most cyanobacteria and chloroplasts their color. Other forms of chlorophyll exist, such as the accessory pigments chlorophyll B, chlorophyll C, chlorophyll D, 12, and chlorophyll F. Chlorophyll B is an olive-green pigment found only in the chloroplasts of plants, green algae, any secondary chloroplasts. Obtained through the secondary endosymbiosis of a green alga, and a few cyanobacteria. 12. It is the chlorophylls A and B together that make most plant and green algal chloroplasts green. 146. Chlorophyll C is mainly found in secondary endosymbiotic chloroplasts that originated from a red alga, although it is not found in chloroplasts of red algae themselves. Chlorophyll C is also found in some green algae and cyanobacteria. 12. Chlorophylls D and F are pigments found only in some cyanobacteria. 12. 147. Carotenoids. In addition to chlorophylls, another group of yellow-orange, 146, pigments called carotenoids are also found in the photosystems. There are about 30 photosynthetic carotenoids. 148. They help transfer and dissipate excess energy. 12. And their bright colors sometimes override the chlorophyll green, like during the fall, when the leaves of some land plants change color. 149. Carotene is a bright red orange carotenoid found in nearly all chloroplasts, like chlorophyll A. 12. Xanthophylls, especially the orange-red zeaxanthin, are also common. 148. Many other forms of carotenoids exist that are only found in certain groups of chloroplasts. 12. Phycobilins. Phycobilins are a third group of pigments found in cyanobacteria, and glaucophyte, red algal, and cryptophyte chloroplasts. 12. 150. Phycobilins come in all colors, though phycoerythrin is one of the pigments that makes many red algae red. 151. Phycobilins often organize into relatively large protein complexes about 40 nanometers across called phycobilisms. 12. Like photosystem I and ATP synthase, phycobilisms jut into the stroma, preventing thylakoid stacking in red algal chloroplasts. 12. Cryptophyte. Chloroplasts and some cyanobacteria don't have their phycobilin pigments organized into phycobilisms, and keep them in their thylakoid space instead. 12. Photosynthetic pigments table of the presence of various pigments across chloroplast groups. Colored cells represent pigment presence. 12. 148, 150. Chlorophyll A, chlorophyll B, chlorophyll C, chlorophyll D, and F. Xanthophylls. Alpha carotene, beta carotene. Phycobilins. Land plants. Green algae. Euglynophytes and chlorarachniophytes. Multicellular red algae. Unicellular red algae. Haptophytes and dinophytes. Paper. Chromatograph. HY of some spinach leaf extract shows the various pigments present in their chloroplasts. Xanthophylls. Chlorophyll A. Chlorophyll B. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Many C4 plants have their mesophyll cells and bundle sheath cells arranged radially around their leaf veins. The two types of cells contain different types of chloroplasts specialized for a particular part of photosynthesis. Cryptophytes, glaucophytes, cyanobacteria. Specialized chloroplasts in C4 plants. See also photosynthesis and C4 photosynthesis. To fix carbon dioxide into sugar molecules in the process of photosynthesis, chloroplasts use an enzyme called rubisco. Rubisco has trouble distinguishing between carbon dioxide and oxygen, so at high oxygen concentrations, rubisco starts accidentally adding oxygen to sugar precursors. This has the result of ATP energy being wasted and CO2 being released, all with no sugar being produced. This is a big problem, since O2 is produced by the initial light reactions of photosynthesis, causing issues down the line in the Calvin cycle, which uses rubisco. 152. C4 plants evolved a way to solve this, by spatially separating the light reactions and the Calvin cycle. The light reactions, which store light energy in ATP and NADP, are done in the mesophyll cells of a C4 leaf. The Calvin cycle, which uses the stored energy to make sugar using rubisco, is done in the bundle sheath cells, a layer of cells surrounding a vein in a leaf. 152. As a result, chloroplasts in C4 mesophyll cells and bundle sheath cells are specialized for each stage of photosynthesis. In mesophyll cells, chloroplasts are specialized for the light reactions, so they lack rubisco, and have normal grana and thylakoids. 133, which they use to make ATP and NADP, as well as oxygen. They store CO2 in a 4-carbon compound, which is why the process is called C4 photosynthesis. The 4-carbon compound is then transported to the bundle sheath chloroplasts, where it drops off CO2 and returns to the mesophyll. Bundle sheath chloroplasts do not carry out the light reactions, preventing oxygen from building up in them and disrupting rubisco activity. 152. Because of this, they lack thylakoids organized into grana stacks, though bundle sheath chloroplasts still have free-floating thylakoids in the stroma, where they still carry out cyclic electron flow, a light-driven method of synthesizing ATP to power the Calvin cycle without generating oxygen. They lack photosystem II, and only have photosystem I, the only protein complex needed for cyclic electron flow. 133, 152, because the job of bundle sheath chloroplasts is to carry out the Calvin cycle and make sugar, they often contain large starch grains. 133. Both types of chloroplast contain large amounts of chloroplast peripheral reticulum, 133, which they use to get more surface area to transport stuff in and out of them. 121, 122, mesophyll chloroplasts have a little more peripheral reticulum than bundle sheath chloroplasts. 153. Location. Distribution in a plant. Not all cells in a multicellular plant contain chloroplasts. All green parts of a plant contain chloroplasts, the chloroplasts, or more specifically, the chlorophyll in them are what make the photosynthetic parts of a plant green. 11. The plant cells which contain chloroplasts are usually parenchyma cells, though chloroplasts can also be found in collenchyma tissue. 154. A plant cell which contains chloroplasts is known as a chlorenchima cell. A typical chlorenchima cell of a land plant contains about 10 to 100 chloroplasts. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. When chloroplasts are exposed to direct sunlight, they stack along the anticlinal cell walls to minimize exposure. In the dark they spread out in sheets along the periclinal walls to maximize light absorption. A cross-section of a leaf, showing chloroplasts in its mesophyll cells. Stomal guard cells also have chloroplasts, though much fewer than mesophyll cells. In some plants such as cacti, chloroplasts are found in the stems, 155, though in most plants, chloroplasts are concentrated in the leaves. 1 square millimeter of leaf tissue can contain 500,000 chloroplasts. 11. Within a leaf, chloroplasts are mainly found in the mesophyll layers of a leaf, and the guard cells of stomata. Palisade mesophyll cells can contain 30–70 chloroplasts per cell, while stomatal guard cells contain only around 8–15 per cell, as well as much less chlorophyll. Chloroplasts can also be found in the bundle sheath cells of a leaf, especially in C4 plants, which carry out the Calvin cycle in their bundle sheath cells. They are often absent from the epidermis of a leaf. 156. Cellular location. Chloroplast movement. See also, cytoplasmic streaming. The chloroplasts of plant and algal cells can orient themselves to best suit the available light. In low light conditions, they will spread out in a sheet, maximizing the surface area to absorb light. Under intense light, they will seek shelter by aligning in vertical columns along the plant cell's cell wall or turning sideways so that light strikes them edge on. This reduces exposure and protects them from photooxidative damage. 157. This ability to distribute chloroplasts so that they can take shelter behind each other or spread out may be the reason why land plants evolved to have many small chloroplasts instead of a few big ones. 158. Chloroplast movement is considered one of the most closely regulated stimulus response systems that can be found in plants. 159. Mitochondria have also been observed to follow chloroplasts as they move. 160. In higher plants, chloroplast movement is run by phototropins, blue light photoreceptors also responsible for plant phototropism. In some algae, mosses, ferns, and flowering plants, chloroplast movement is influenced by red light in addition to blue light. 157, though very long red wavelengths inhibit movement rather than speeding it up. Blue light generally causes chloroplasts to seek shelter, while red light draws them out to maximize light absorption. 160. Studies of Vallisneria gigantea, an aquatic flowering plant, have shown that chloroplasts can get moving within 5 minutes of light exposure, though they don't initially show any net directionality. They may move along microfilament tracks, and the fact that the microfilament mesh changes shape to form a honeycomb structure surrounding the chloroplasts after they have moved suggests that microfilaments may help to anchor chloroplasts in place. 159, 160. Function and chemistry. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Guard cell chloroplasts. This section needs expansion with determined Functions, controversial. Functions, characteristics and population. You can help by adding to it. August 2013. Unlike most epidermal cells, the guard cells of plant stomata contain relatively well-developed chloroplasts. 156, however, exactly what they do is controversial. 161. Plant innate immunity. Plants lack specialized immune cells. All plant cells participate in the plant immune response. Chloroplasts, along with the nucleus, cell membrane, and endoplasmic reticulum, 162, are key players in pathogen defense. Due to its role in a plant cell's immune response, pathogens frequently target the chloroplast. 162. Plants have two main immune responses: the hypersensitive response, in which infected cells seal themselves off and undergo programmed cell death, and systemic acquired resistance, where infected cells release signals warning the rest of the plant of a pathogen's presence. Chloroplasts stimulate both responses by purposely damaging their photosynthetic system, producing reactive oxygen species. High levels of reactive oxygen species will cause the hypersensitive response. The reactive oxygen species also directly kill any pathogens within the cell. Lower levels of reactive oxygen species initiate systemic acquired resistance, triggering defense molecule production in the rest of the plant. 162. In some plants, chloroplasts are known to move closer to the infection site and the nucleus during an infection. 162. Chloroplasts can serve as cellular sensors. After detecting stress in a cell, which might be due to a pathogen, chloroplasts begin producing molecules like salicylic acid, jasmonic acid, nitric oxide and reactive oxygen species which can serve as defense signals. As cellular signals, reactive oxygen species are unstable molecules, so they probably don't leave the chloroplast, but instead pass on their signal to an unknown second messenger molecule. All these molecules initiate retrograde signaling, signals from the chloroplast that regulate gene expression in the nucleus. 162. In addition to defense signaling, chloroplasts, with the help of the peroxisms, 163, help synthesize an important defense molecule, jasmonate. Chloroplasts synthesize all the fatty acids in a plant cell. 162, 164, linoleic acid, a fatty acid, is a precursor to jasmonate. 162. Photosynthesis. Main article, photosynthesis. One of the main functions of the chloroplast is its role in photosynthesis, the process by which light is transformed into chemical energy, to subsequently produce food in the form of sugars. Water, H2O, and carbon dioxide, CO2, are used in photosynthesis, and sugar and oxygen, O2, are made, using light energy. Photosynthesis is divided into two stages: the light reactions, where water is split to produce oxygen, and the dark reactions, or Calvin's cycle, which builds sugar molecules from carbon dioxide. The two phases are linked by the energy carriers. Adenosine triphosphate, ATP, and nicotinamide adenine dinucleotide phosphate, NADP+. 165, 166. Light reactions. Main article, light reactions. The light reactions take place on the thylakoid membranes. They take light energy and store it in NADP, a form of NADP+, and ATP to fuel The dark reactions. Energy carriers. Main articles, adenosine triphosphate and NADPH. ATP is the phosphorylated version of adenosine diphosphate, ADP, which stores energy in a cell and powers most cellular activities. ATP is the energized form, while ADP is the partially depleted form. NADPH is an electron carrier which ferries high-energy electrons. In the light reactions, it gets reduced, meaning it picks up electrons, becoming NADPH. Photophosphorylation. Main article, Photophosphorylation. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. The light reactions of photosynthesis take place across the thylakoid membranes. The Calvin cycle, interactive diagram. The Calvin cycle incorporates carbon dioxide into sugar molecules. Like mitochondria, chloroplasts use the potential energy stored in an H+ or hydrogen ion gradient to generate ATP energy. The two photosystems capture light energy to energize electrons taken from water, and release them down an electron transport chain. The molecules between the photosystems harness the electrons' energy to pump hydrogen ions into the thylakoid space, creating a concentration gradient, with more hydrogen ions, up to 1,000 times as many, 112, inside the thylakoid system than in the stroma. The hydrogen ions in the thylakoid space then diffuse back down their concentration gradient, flowing back out into the stroma through ATP synthase. ATP synthase uses the energy from the flowing hydrogen ions to phosphorylate adenosine diphosphate into adenosine triphosphate, or ATP. 112, 167. Because chloroplast ATP synthase projects out into the stroma, the ATP is synthesized there, in position to be used in the dark reactions. 168. NADPH reduction. See also, redox reaction. Electrons are often removed from the electron transport chains to charge NADPH with electrons, reducing it to NADPH. Like ATP synthase. Ferredoxin NADPH reductus, the enzyme that reduces NADPH, releases the NADPH it makes into the stroma, right where it is needed for the dark reactions. 168. Because NADPH reduction removes electrons from the electron transport chains, they must be replaced, the job of photosystem 2, which splits water molecules, H2O, to obtain the electrons from its hydrogen atoms. 112, 165. Cyclic photophosphorylation. Main article, cyclic photophosphorylation. While photosystem 2 photolyzes water to obtain and energize new electrons, photosystem I simply reenergizes depleted electrons at the end of an electron transport chain. Normally, the reenergized electrons are taken by NADPH, though sometimes they can flow back down more H+ pumping electron transport chains to transport more hydrogen ions into the thylakoid space to generate more ATP. This is termed cyclic photophosphorylation because the electrons are recycled. Cyclic photophosphorylation is common in C4 plants, which need more ATP than NADPH. 152. Dark reactions. Main article, dark reactions. The Calvin cycle, also known as the dark reactions, is a series of biochemical reactions that fixes CO2 into G3P sugar molecules and uses the energy and electrons from the ATP and NADF made in the light. Reactions. The Calvin cycle takes place in the stroma of the chloroplast. 152. While named the dark reactions, in most plants, they take place in the light, since the dark reactions are dependent on the products of the light. Reactions. 11. Carbon fixation and G3P synthesis. The Calvin cycle starts by using the enzyme rubisco to fix CO2 into 5-carbon-ribulose bisphosphate, RUBP, molecules. The result is unstable 6-carbon molecules that immediately break down into 3-carbon molecules called 3-phosphoglyceric acid, or 3-PGA. The ATP and NADF made in the light reactions is used to convert the 3-PGA into glyceraldehyde 3-phosphate, or G3P sugar molecules. Most of the G3P molecules are recycled back into RUBP using energy from more ATP, but 1 out of every 6 produced leaves the cycle, the end product of the dark reactions. 152. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Sucrose is made up of a glucose monomer, left, and a fructose monomer, right. Sugars and starches. Glyceraldehyde 3-phosphate can double up to form larger sugar molecules like glucose and fructose. These molecules are processed, and from them, the still larger sucrose, a disaccharide commonly known as table sugar, is made, though this process takes place outside of the chloroplast, in the cytoplasm. 169. Alternatively, glucose monomers in the chloroplast can be linked together to make starch, which accumulates into the starch grains found in the chloroplast. 169. Under conditions such as high atmospheric CO2 concentrations, these starch grains may grow very large, distorting the grana and thylakoids. The starch granules displace the thylakoids, but leave them intact. 170. Waterlogged roots can also cause starch buildup in the chloroplasts, possibly due to less sucrose being exported out of the chloroplast, or more accurately, the plant cell. This depletes a plant's free phosphate supply, which indirectly stimulates chloroplast starch synthesis. 170. While linked to low photosynthesis rates, the starch grains themselves may not necessarily interfere significantly with the efficiency of photosynthesis. 171, and might simply be a side effect of another photosynthesis depressing factor. 170. Photorespiration. Photorespiration can occur when the oxygen concentration is too high. Rubisco cannot distinguish between oxygen and carbon dioxide very well, so it can accidentally add O2 instead of CO2 to RUBP. This process reduces the efficiency of photosynthesis, it consumes ATP and oxygen, releases CO2, and produces no sugar. It can waste up to half the carbon fixed by the Calvin cycle. 165. Several mechanisms have evolved in different lineages that raise the carbon dioxide concentration relative to oxygen within the chloroplast, increasing the efficiency of photosynthesis. These mechanisms are called carbon dioxide concentrating mechanisms, or CCMs. These include crassulation acid metabolism, C4 carbon fixation, 165, and pyrenoids. Chloroplasts in C4 plants are notable as they exhibit a distinct chloroplast dimorphism. pH Because of the H and gradient across the thylakoid membrane, the interior of the thylakoid is acidic, with a pH around 4,172, while the stroma is slightly basic, with a pH of around 8,173. The optimal stroma pH for the Calvin cycle is 8.1, with the reaction nearly stopping when the pH falls below 7.3. 174. CO2 in water can form carbonic acid, which can disturb the pH of isolated chloroplasts, interfering with photosynthesis, even though CO2 is used in photosynthesis. However, chloroplasts in living plant cells are not affected by this as much. 173. Chloroplasts can pump K and H and ions in and out of themselves using a poorly understood light-driven transport system. 173. In the presence of light, the pH of the thylakoid lumen can drop up to 1.5 pH units, while the pH of the stroma can rise by nearly 1 pH unit. 174. Amino acid synthesis. Chloroplasts alone make almost all of a plant cell's amino acids in their stroma, 175, except the sulfur-containing ones like cysteine and methionine. 176, 177. Cysteine is made in the chloroplast, the proplastide too, but it is also synthesized in the cytosol and mitochondria. Probably because it has trouble crossing membranes to get to where it is needed. 177. The chloroplast is known to make the precursors to methionine, but it is unclear whether the organelle carries out the last leg of the pathway or if it happens in the cytosol. 178. Other nitrogen compounds. Chloroplasts make all of a cell's purines and pyrimidines, the nitrogenous bases found in DNA and RNA. 175. They also convert nitrite NO2 into ammonia, NH3, which supplies the plant with nitrogen to make its amino acids and nucleotides. 175. Other chemical products. This section needs expansion with needs more. About lipids, also paramlin. You can help by adding to it. March 2013. The plastide is the site of diverse and complex lipid synthesis in plants. 179, 180. The carbon used to form the majority of the lipid is from acetyl-CoA, which is the decarboxylation product of pyruvate. 179. Pyruvate may enter the plastide from the cytosol by passive diffusion. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Plastide types, interactive diagram, plants contain many different kinds of plastids in their cells. Many plastide interconversions are possible through the membrane after production in glycolysis. 181. Pyruvate is also made in the plastide from phosphenylpyruvate, a metabolite made in the cytosol from pyruvate or PGA. 179. Acetate in the cytosol is unavailable for lipid biosynthesis in the plastide. 182. The typical length of fatty acids produced in the plastide are 16 or 18 carbons, with 0 to 3 cis double bonds. 183. The biosynthesis of fatty acids from acetyl-CoA primarily requires two enzymes. Acetyl-CoA carboxylase creates malonyl-CoA, used in both the first step and the extension steps of synthesis. Fatty acid synthase, FAS, is a large complex of enzymes and cofactors including acyl carrier protein, ACP, which holds the acyl chain as it is synthesized. The initiation of synthesis begins with the condensation of malonyl ACP with acetyl-CoA to produce ketobutyryl ACP. 2 reductions involving the use of NADPH and 1 dehydration creates butyryl ACP. Extension of the fatty acid comes from repeated cycles of malonyl ACP condensation, reduction, and dehydration. 179. Other lipids are derived from the methylerythritol phosphate, MEP, pathway and consist of gibberellins, sterols, abscisic acid, phytol, and innumerable secondary metabolites. 179. Differentiation, replication, and inheritance. Main article, plastide. Chloroplasts are a special type of a plant cell organelle called a plastide, though the two terms are sometimes used interchangeably. There are many other types of plastids, which carry out various functions. All chloroplasts in a plant are descended from undifferentiated proplastids, found in the zygote, 175, or fertilized egg. Proplastids are commonly found in an adult plants. Apical meristums. Chloroplasts do not normally develop from proplastids in root tip. Meristums, 184, instead, the formation of starch-storing amyloplasts is more common. 175. In shoots, proplastids from shoot apical meristums can gradually develop into chloroplasts in photosynthetic leaf tissues as the leaf matures, if exposed to the required light. 15. This process involves invaginations of the inner plastide membrane, forming sheets of membrane that project into the internal stroma. These membrane sheets then fold to form thylakoids and grana. 185. If angiosperm shoots are not exposed to the required light for chloroplast formation, proplastids may develop into an etioplast stage before becoming chloroplasts. An etioplast is a plastide that lacks chlorophyll, and has inner membrane invaginations that form a lattice of tubes in their stroma, called a prolamellar body. While etioplasts lack chlorophyll, they have a yellow chlorophyll precursor stocked. 15. Within a few minutes of light exposure, the prolamellar body begins to reorganize into stacks of thylakoids, and chlorophyll starts to be produced. This process, where the etioplast becomes a chloroplast, takes several hours. 185. Gymnosperms do not require light to form chloroplasts. 185. Light, however, does not guarantee that a proplastide will develop into a chloroplast. Whether a proplastide develops into a chloroplast some other kind of plastide is mostly controlled by the nucleus, 15, and is largely influenced by the kind of cell it resides in. 175. Plastide interconversion. Plastide differentiation is not permanent, in fact many interconversions are possible. Chloroplasts may be converted to chromoplasts, which are pigment-filled plastids responsible for the bright colors seen in flowers and ripe fruit. Starch-storing amyloplasts can also be converted to chromoplasts, and it is possible for proplastids to develop straight into chromoplasts. Chromoplasts and amyloplasts can also become chloroplasts, like what happens when a carrot or a potato is illuminated. If a plant is injured, or something else causes a plant cell to revert to a meristematic state, chloroplasts and other plastids can turn back into proplastids. Chloroplast, amyloplast, chromoplast, proplastide are not absolute state. Intermediate forms are common. 175. Division. This section needs expansion with functions, Z. Ring dynamic assembly. Regulators such as giant. Chloroplast 1. You can help explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF by adding to it. February 2013. Most chloroplasts in a photosynthetic cell do not develop directly from proplastids or etioplasts. In fact, a typical shoot meristematic plant cell contains only 720 proplastids. These proplastids differentiate into chloroplasts, which divide to create the 30-70 chloroplasts found in a mature photosynthetic plant cell. If the cell divides, chloroplast division provides the additional chloroplasts to partition between the two daughter cells. 186. In single-celled algae, chloroplast division is the only way new chloroplasts are formed. There is no proplastide differentiation. When an algal cell divides, its chloroplast divides along with it, and each daughter cell receives a mature chloroplast. 185. Almost all chloroplasts in a cell divide, rather than a small group of rapidly dividing chloroplasts. 187. Chloroplasts have no definite S phase. Their DNA replication is not synchronized or limited to that of their host cells. 188. Much of what we know about chloroplast division comes from studying organisms like Arabidopsis and the red alga Cyanidea meroli. 158. Most chloroplasts in plant cells, and all chloroplasts in algae arise from chloroplast division. 185. Picture references, 158, 189. The division process starts when the proteins FTSZ1 and FTSZ2 assemble into filaments, and with the help of a protein arc-6, form a structure called a Z-ring within the chloroplast stroma. 158, 189. The MIN system manages the placement of the Z-ring, ensuring that the chloroplast is cleaved more or less evenly. The protein mind prevents FTSZ from linking up and forming filaments. Another protein arc-3 may also be involved, but it is not very well understood. These proteins are active at the poles of the chloroplast, preventing Z-ring formation there, but near the center of the chloroplast, MIN inhibits them, allowing the Z-ring to form. 158. Next, the two plastide dividing rings, or PD rings form. The inner plastide dividing ring is located in the inner side of the chloroplast's inner membrane, and is formed first. 158. The outer plastide dividing ring is found wrapped around the outer chloroplast membrane. It consists of filaments about 5 nanometers across, 158, arranged in rows 6.4 nanometers apart, and shrinks to squeeze the chloroplast. This is when chloroplast constriction begins. 189. In a few species like Cyanidea meroli, chloroplasts have a third plastide dividing ring located in the chloroplast's intermembrane space. 158, 189. Late into the constriction phase, dynamin proteins assemble around the outer plastide dividing ring, 189, helping provide force to squeeze the chloroplast. 158. Meanwhile, the Z-ring and the inner plastide dividing ring break down. 189. During this stage, the many chloroplast DNA Plasmids floating around in the stroma are partitioned and distributed to the two forming daughter chloroplasts. 190. Later, the dynamins migrate under the outer plastide dividing ring, into direct contact with the chloroplast's outer membrane, 189, to cleave the chloroplast in two daughter chloroplasts. 158. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. Chloroplast division in this light micrograph of some moss chloroplasts, many dumbbell-shaped chloroplasts can be seen dividing. Grana are also just barely visible as small granules. A remnant of the outer plastide dividing ring remains floating between the two daughter chloroplasts, and a remnant of the dynamin ring remains attached to one of the daughter chloroplasts. 189. Of the 5 or 6 rings involved in chloroplast division, only the outer plastide dividing ring is present for the entire constriction and division phase, while the Z ring forms first. Constriction does not begin until the outer plastide dividing ring forms. 189. Regulation. In species of algae that contain a single chloroplast, regulation of chloroplast division is extremely important to ensure that each daughter cell receives a chloroplast. Chloroplasts can't be made from scratch. 83, 158. In organisms like plants, whose cells contain multiple chloroplasts, coordination is looser and less important. It is likely that chloroplast and cell division are somewhat synchronized, though the mechanisms for it are mostly unknown. 158. Light has been shown to be a requirement for chloroplast division. Chloroplasts can grow and progress through some of the constriction stages under poor quality green light, but are slow to complete division. They require exposure to bright white light to complete division. Spinach leaves grown under green light have been observed to contain many large dumbbell-shaped chloroplasts. Exposure to white light can stimulate these chloroplasts to divide and reduce the population of dumbbell-shaped chloroplasts. 187, 190. Chloroplast inheritance. Like mitochondria, chloroplasts are usually inherited from a single parent. Biparental chloroplast inheritance, where plastide genes are inherited from both parent plants, occurs in very low levels in some flowering plants. 191. Many mechanisms prevent biparental chloroplast DNA inheritance, including selective destruction of chloroplasts or their genes within the gamete or zygote, and chloroplasts from one parent being excluded from the embryo. Parental chloroplasts can be sorted so that only one type is present in each offspring. 192. Gymnosperms, such as pine trees, mostly pass on chloroplasts paternally. 193, while flowering plants often inherit chloroplasts maternally. 194, 195. Flowering plants were once thought to only inherit chloroplasts maternally. However, there are now many documented cases of angiosperms inheriting chloroplasts paternally. 191. Angiosperms, which pass on chloroplasts maternally, have many ways to prevent paternal inheritance. Most of them produce sperm cells that do not contain any plastids. There are many other documented mechanisms that prevent paternal inheritance in these flowering plants, such as different rates of chloroplast replication within the embryo. 191. Among angiosperms, paternal chloroplast inheritance is observed more often in hybrids than in offspring from parents of the same species. This suggests that incompatible hybrid genes might interfere with the mechanisms that prevent paternal inheritance. 191. Transblastomic plants. Recently, chloroplasts have caught attention by developers of genetically modified crops. Since, in most flowering plants, chloroplasts are not inherited from the male parent, transgenes in these plastids cannot be disseminated by pollen. This makes plastide transformation a valuable tool for the creation and cultivation of genetically modified plants that are biologically contained, thus posing significantly lower environmental risks. This biological containment strategy is therefore suitable for establishing the coexistence of conventional and organic agriculture. While the reliability of this mechanism has not yet been studied for all relevant crop species, recent results in tobacco plants are promising, showing a failed containment rate of transblastomic plants at 3 in 1 million. 195. Footnotes. 1 AB not to be confused with chromatophore, the pigment cells in some animals, or chromatophore, the membrane-associated vesicle in some bacteria. 2 For this reason, glaucophyte chloroplasts are also known as muroplasts from the Latin muro meaning wall. References. Image times labels. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 1 Jones D. 2003, 1917. Roach P., Hartman J., Setter J., eds. English Pronouncing Dictionary. Cambridge, Cambridge University Press. ISBN 3-12-539683-2. 2 Chloroplast. Merriam-Webster.com Dictionary. Merriam-Webster. 3 Basic Biology, the 18th of March, 2016. Bacteria. 4 von Moll, H. 1835/1837. Über die Vermehrung der Pflanzen. Zellendurchteilung. Dessert. Tübingen 1835. Flora 1837. 5 AB Schimper, Af. 1883. Über die Entwicklung der Chlorophyllkerne und Farbe, about the development of the chlorophyll grains and stains. Bot. Zeitung, in German. 41, 10514. 12131, 13746, 15362. Archived from the original on 19 October 2013. 6 Strasburger E., 1884. Das Botanische Praktikum, 1st ed. Jena. Gustav Fischer. 7 Gunning B., Koenig F., Gavin G.P., 2006. A dedication to pioneers of research on chloroplast structure. In Wise R. Huber. J.K., eds. The Structure and Function of Plastids. Netherlands. Springer. pp. 23 xxi. ISBN 9781402065705. 8 Huber J.K., 1984. Chloroplasts. New York, Plenum. ISBN 9781461327677. 9 Chloroplast. Online Etymology Dictionary. 10 More Kr. Magnabisco C. Momper L. Gold the. Basark T. Fornia. GP, 2019. An expanded ribosomal phylogeny of cyanobacteria. Supports a deep placement of plastids. Frontiers in Microbiology. 10, 1612. Do. 10.3389/fmic.2019.01612. PMC 66402009. PMID 31354692. 11 ABCDE Campbell Na, Rees JB, Yori LA, Kane ML, Wasserman. Mineski PV, Jackson RB, 2009. Biology, 8th ed. Benjamin. Cummings, Pearson. pp. 186187. ISBN 978-0-8053-6844-4. 12 ABCDEFGHIJKLMNOPQRSTUVWXYZAAABACADAFACARIAJ. Ak Elimineo Apacaras at Kimi, Archibald J.M., 2009. Diversity and Evolution of Plastids and Their Genomes. In Sandelius as Aronson H., eds. The Chloroplast. Plant Cell Monographs. Volume 13. pp. 139. Do. 10.1007835406869665_1. ISBN 978-3-540. 68692-7. S2 SID 83672683. 13 Bryant The, Gulilmiji, Damasik NT, Karstetsum, Cohen Baziah. G. 1979. The Structure of Cyanobacterial Phacobolisms, a Model. Archives of Microbiology. 123, 231134. Bibcode, 1979 ARMIC.123113b. Do. 10.1007/BF00446810. S2 SID 1589428. 14 Miresk-Kowalski K. 1905. Über Nature und Ursprung der Chromatophoren im Pflanzenreich, about the nature and origin of chromatophores in the vegetable kingdom. By All Centralbel, in German. 25, 593604. 15 ABCD Alberts B. 2002. Molecular Biology of the Cell, 4 ed. New York, UA, Garland. ISBN 0-8153-4072-9. 16 Gaber, Grossman R., Botticheria D., August 2020. Paulinella. A Model for Understanding Plastide Primary Endosymbiosis. J. Facol. 56, 48378843. Bibcode, 2020 JPCGY 56837G. Do. 10.1011/JPY.13003. PMC 77348444. PMID 322898979. 17 ABCDEFGHIJ Campbell Na, Rees JB, Yori LA, Kane ML. Wasserman, Mineski PV, Jackson RB, 2009. Biology, 8th ed. Benjamin Cummings, Pearson. p. 516. ISBN 978-0-8053-6844-4. 18 ABCDE Milo R. Phillips R. Cell Biology by the Numbers, How Large Are Chloroplasts? Book.bionumbers.org. Retrieved 7 February 2017. 19 A.B. Sanchez Baracaldo P., Raven J.A., Pi Sunny D., Noel R. September 2017. Early photosynthetic eukaryotes inhabited low salinity habitats. Proceedings of the National Academy of Sciences of the United States of America. 114. 37 E 7737 E 7745. Bibcode 2017 as 114 E 7737 S. Do 10.1070/NAS.1620089114. PMC 5603991. PMID 2880807. 20 Falcon, Louisa I., Magellan, Susanna, Castillo, Amanda, 4 March 2010. Dating the cyanobacterial ancestor of the chloroplast. The ISM Journal 467777883. Bibcode 2010 ISMEJ 4777F. Do 10.1038/ISMEJ.2010.2. ISSN 1751-7362. PMID 202000567. 21 ABCDE Nakia Marti, Archibald J.M., April 2012. Evolving a photosynthetic organelle. BMC Biology. 10.135. Do 10.1141-7741-7007-10-35. PMC 3337241. PMID 225312110. 22 ABCD McFadden G.I., January 2001. Chloroplast Origin and Integration. Plant Physiology. 125, 153. Do 10.1104/pp.125.1.50. PMC 1539323. PMID 11154294. 23 ABCDEFGHIJKLMNOPQRSTYSOR, Huber J.K., 2006. The Structure and Function of Plastids. Dordrecht, Springer. pp. 321. ISBN 978-1-4020-4061-0. Archived from the original on 8 March 2016. Retrieved 21 May 2013. 24 Ponce Toledo I., Deschamps P., Lopez Garcia P., Zivanovic Y. Benzareira K., Moreira D., February 2017. An early branching freshwater cyanobacterium at the origin of plastids. Current Biology 27, 3386-391. Bibcode 2017 CBio 27386P. Do 10.1016/J.Cub.2016.11.056. PMC 5650054. PMID 28132810. 25 De Vries J., Archibald J.M., February 2017. Endosymbiosis, did. Plastids evolved from a freshwater cyanobacterium. Current biology. 27, 3R103R105. Bibcode 2017 CBio 27.R103D. Do 10.1000.j.cub.2016.12.006. PMID 28171752. 26 Lopez Garcia P., Imel, Moreira D., December 2017. Symbiosis in Eukaryotic Evolution. Journal of Theoretical Biology. 434, 20. 33 Bibcode 2017 JTB.434, 20L. Do 10.1016/j.tb.2017.02.031. PMC 5638015. PMID 28254477. 27 AB Macarano, Luis, Noack, Eva C.M. 13 September 2021. Paulinella chromatophora. Current biology. 31, 17 R1024. R1026. Do 10.1016/j.cub.2021.07.028. 28 Archibald J.M., January 2009. The Puzzle of Plastide Evolution. Current biology. 19, 2R81-8. Bibcode 2009 CBio 19R810. Do 10.1016/j.cub.2008.11.067. PMID 19174147. S2 SID 51989. 29 ABCDEFGHIJKLMNOPQRSTUVWXYZAAAB Keeling PJ, October 2004. Diversity and Evolutionary History of Plastids and Their Hosts. American Journal of Botany. 91, 10.1481-93. Do 10.3732/ACHP.91.10.1481. PMID 21652304. S2 SID 17522125. 30 McFadden, Jeffrey I. Van Doren, Guile G. 2004. Evolution. Red algal genome affirms a common origin of all plastids. Current biology. 14, 13 R514 R516. Bibcode 2004 CBio 14.R514M. Do 10.1016/j.cub.2004.06.041. PMID 15242632. 31 ABCDEFGHIJKLMNOPQRSTUVWXYZAAABAC Keeling PJ. March 2010. The Endosymbiotic Origin, Diversification and Fate of Plastids. Philosophical Transactions of the Royal Society of London. Series B, Biological Sciences. 365, 1541 72948. Do 10.1098/RSTB.2009.0103. PMC 28172223. PMID 20124341. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 32 A Bika BK, Green BR, February 2005. Protein import pathways. In complex chloroplasts derived from secondary endosymbiosis, involving a red algal ancestor. Plant molecular biology. 57, 3333. 42 do, 10.1007/4-7848Y. PMID 158300125. S2 SID 226190029. 33 Ball S, Kalioni C, Sansai U, Raj JN, 30 C, March 2011. The evolution of glycogen and starch metabolism in eukaryotes gives molecular clues to understand the establishment of plastide endosymbiosis. Journal of Experimental Botany. 62, 1775. 801 do, 10.1093/JXB/ERC411. PMID 21220783. 34 Guiry, Michael D. 2 January 2024. How many species of algae are there? A reprise. 4 kingdoms, 14 phyla, 63 classes and still growing. Journal of Phycology. 60, 2214228. Bib code, 2024 JPCGY 6214G. Do, 10.1111/JPY.13431. ISSN 0022-3646. PMID 38245909. 35 Archibald, John M. 27 January 2009. The Puzzle of Plastide. Evolution. Current Biology. 19, 2R81R88. Bib code, 2009 C-Bio, 19R81O. Do, 10.1016/J.Cub.2008.11.067. PMID 19174147. 36 Mijishima, Shinya, 1 March 2011. Mechanism of plastide division, from a bacterium to an organelle. Plant Physiology. 155, for 1533 1544 do, 10.1104/pp.110.170688. ISSN 1532. 2548. PMC 3091088. PMID 21311032. 37 Wise, Robert R. Huber, J. Kenneth, eds. 2006. The Structure and Function of Plastids. Advances in Photosynthesis and Respiration. Volume 23. Dordrecht, Springer, Netherlands. Do, 10.1007/4-978-1-4020-4061-0. ISBN 978-1-4020-4060-3. 38 Rodo. The Free Dictionary. Farlex. Retrieved 7 June 2013. 39 ABCDEF Campbell Na, Rees JB, Yoi LA, Kane ML, Wasserman. Mineski PV, Jackson RB, 2009. Biology, 8th ed. Benjamin. Cummings, Pearson. pp. 58292. ISBN 978-0-8053-6844-4. 40 A.B. Lewis L.A., McCourt R.M., October 2004. Green Algae and the Origin of Land Plants. American Journal of Botany. 91, 10 1535 56. Do. 10.3091.10.1535. PMID 216523008. 41 Makida M, Take Ik, Sato H, Chung S.J., Kuroiwa H, Takeo S. It al. April 2006. Genes for the peptidoglycan synthesis pathway are essential for chloroplast division in moss. Proceedings of the National Academy of Sciences of the United States of America. 103. 17 6753 8. Bib code 2006 NAS 103.6753 M. Do. 10.1073/NAS.0510693103. PMC 1458953. PMID 166189224. 42 Morani J.V., Samachia, January 1999. How do algae concentrate CO2 to increase the efficiency of photosynthetic carbon fixation? Plant Physiology. 119, 1916. Do. 10.1104/pp.119.1.9. PMC 1539202. PMID 9880340. 43 Tartara, Busias D.G., April 2004. The non-photosynthetic pathogenic green alga Helicosporidium sp. has retained a modified functional plastide genome. FEMS Microbiology Letters. 233, 1. 1537. Do. 10.1016/J.FEMSLE.2004.02.006. PMID 150438882. 44 ABC Noack E.C., Fogel H., Groth M., Grossman R., Melkonian M. Glockner G., January 2011. Endosymbiotic gene transfer and transcriptional regulation of transferred genes in Paulinella. Chromatophora. Molecular Biology and Evolution. 28, 140722. Do. 10.1093/Molbev/MSQ209. PMID 20702568. 45 Archibald, John M. The 25th of September, 2017. Evolution, Protein. Import in a nascent photosynthetic organelle. Current Biology. 27. 18 R1004 R1006 Do. 10.1016/J.Cub.2017.08.013. ISSN 0960 9822. 46 Singer, Anna, Postman, Dron, Mullick, Cornelia, Valadez. Cano, Cecilio, Hansk, Sebastian, Huron, Vanessa, Rensing, Stefan. Adot, Stuller, Kai, Noack, Eva CM. The 25th of September, 2017. Massive protein import into the early evolutionary stage photosynthetic organelle of the amoeba Paulinella chromatophora. Current biology 27, 18 2763 2773.E5 to 10.1016/j.cub.2017.08.010 ISSN 0960-9822 47 AB Mustafa, Bestari B, Maya UG, Bola C, Valentin K. Bottasheria D, June 2009. Genomic footprints of a cryptic plastide. Endosymbiosis in diatoms, PDF. Science. 324, 5935 1724 6. Bibcode, 2009 SCI, 324.1724 M. Do, 10.1026/science.1172983. PMID 19556510. S2 SID 11408339. 48 Rogers MB, Gulson PR, Sue V, McFadden GI, Keeling PJ, January 2007. The Complete Chloroplast Genome of the Chlorarachniophyte. By Giloila Natans, Evidence for Independent Origins of Chlorarachniophyte and Euglynid Secondary Endosymbionts. Molecular Biology and Evolution. 24, 15462. Do. 10.1093/Molbev/MSL129. PMID 169904339. 49 ABCDEFGHIJKLMN Hackett JD, Anderson DM, Erdner DL. Bottasheria D, October 2004. Dinoflagellates, a remarkable evolutionary experiment. American Journal of Botany. 91, 10. 1523-34. Do. 10.3791.10.1523. PMID 21652307. 50 Toledo I, the 5th of March, 2018. Origins and Early Evolution of Photosynthetic Eukaryotes, Thesis. Universite Paris Saclay. 51 Bodilla, February, 2018. Did some red alga-derived plastids evolve via kleptoplastidae? A hypothesis. Biological Reviews of the Cambridge Philosophical Society. 93, 1201222. Do. 10.1111/BRV.12340. PMID 285444184. S2 SID 2461383. 52 AB Janukovic J, Gavalis GS, Berkey F, Din D, Barkvarov TR. Gornick SG, IT al. January 2017. Major transitions in dinoflagellate evolution unveiled by Philotranscriptomics. Proceedings of the National Academy of Sciences of the United States of America. 114. 2E171E180. Bibcode 2017 NAS 114E.171J. Do. 10.1073/NAS.16148421114. PMC 52407007. PMID 280282238. 53 ABCD NER SC, Strippen B, August 2011. What do human parasites do with a chloroplast anyway? PLOS Biology. 9, 8. E10011137. Do. 10.1371/journal.bio.10011137. PMC 31666169. PMID 219125515. 54 Quigger, Kotabova E, Cherov J, Kawa, Setlik J, Sediva B, it al. 10 October 2012. Photosynthesis in chromarovalia represents a simple system with high efficiency. PLOS 1. 7, 10E47036. Bibcode 2012 PLOSO 747036Q. Do. 10.1371/journal.pone.0047036. PMC 34684883. PMID 23071705. 55 Doral RG, Smith AG, July 2011. Do red and green make brown. Perspectives on plastide acquisitions within chromalveolates. Eukaryotic cell. 10, 7856668. Do. 10.1111/EC.00326-10. PMC 31474421. PMID 216222904. 56 Meeson BW, Chang SS, Sweeney BM, 1982. Characterization of peridine in chlorophyll proteins from the marine dinoflagellate. Kiratium furca. Botanica marina. 25, 834750. Do. 10.15/bottom.1982.25.8.347. S2 SID 838671003. 57 ABC Tengs T, Dahlbergo J, Schulchian Tabrizi K, Klavnus D, Rudy K, Delwich CF, Jacobson KS, May 2000. Phylogenetic analyses indicate that the 19-hexanoyloxyfucoxanthin containing dinoflagellates have tertiary plastids of haptophyte origin. Molecular biology and evolution. 17, 571829. Do. 10.101934/oxfordjournals.molbev.0263550. PMID 10779532. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 58 ABC Schnepf, Elbrachter M, 1999. Dinophyte chloroplasts and Phylogenia Review. Grana. 38, 238197. Bibcode, 1999 Grana 38, 81S. Do. 10.1080/001731399085592217. 59 Skofgård, 1998. Role of chloroplast retention in a marine dinoflagellate. Aquatic microbial ecology. 15, 293301. Do. 10.3054/AME015293. 60 Doral RG, Howe CJ, August 2015. Integration of plastids with their hosts, Lessons learned from dinoflagellates. Proceedings of the National Academy of Sciences of the United States of America. 112, 3310244754. Bib code 2015 NAS 112102447D. Do 10.1073 NAS.14213801112. PMC 4547248. PMID 259953666. 61 Carat Stocking, CR. And Gifford, E.M. 1959. Incorporation of thymidine into chloroplasts of Spirogyra. Biochemical and Biophysical Research Communications. 1. 3159164 do 10.1016/0006 291x 59 90010-5 62 Carat Ris, H. Plot, W. 1962. Ultrastructure of DNA containing areas in the chloroplast of Chlamydomonas. J. Cell Biol. 13, 338391. Do 10.1083/JCB.13.3.383. PMC 2106071. PMID 1449246. 63 Carat Lyttelton, J.W. 1962. Isolation of ribosomes from spinach chloroplasts. EXP. Cell Res. 26, 13123117 do 10.1016/0014. 4827, 62, 90183-0. PMID 144678684. 64 Carat Heber, U. 1962. Protein synthesis in chloroplasts during photosynthesis. Nature. 195, 191-92. Bib code 1962 Nature.195, 91H. Do 10.1038/1950910. PMID 139058112. S2 SID 4265095. 65 Carat Chloroplasts and Other Plastids. University of Hamburg. Archived from the original on 25 September 2012. Retrieved 27 December 2012. 66 Carat ABCDEFG Sundali Usas, 2009. The Chloroplast, Interactions with the Environment. Springer. P18. ISBN 978-3-540-68696-5. 67 Carat ABC Dan L., 2002. Bioscience, Explained, PDF. Green DNA. Bioscience Explained. Archived, PDF, from the original on 14 December 2010. 68 Carat ABCD Clegg Mount, Gort B.S., Learn G.H., Morton B.R., July 1994. Rates and Patterns of Chloroplast DNA Evolution. Proceedings of the National Academy of Sciences of the United States of America. 91, 15 6795 801. Bib code 1994 NAS 91.6795 C. Do 10.1073/91.15.6795. PMC 44285. PMID 8041699. 69 ABCD Shaw J, Licky EB, Schilling EE, Small RL, March 2007. Comparison of whole chloroplast genome sequences to choose non-coding regions for phylogenetic studies in angiosperms, the tortoise and the hare 3. American Journal of Botany. 94, 3275. 88 do. 10.3732/hp.94.3.275. PMID 216366401. S2 SID 305011148. 70 AB Burgess J, 1989. An Introduction to Plant Cell Development. Cambridge, Cambridge University Press. p. 62. ISBN 0-521-31611-1. 71 Green, Beverly R. 28 April 2011. Chloroplast genomes of photosynthetic eukaryotes. The Plant Journal. 66, 134-44. Do. 10.1011/J.1003X.2011.04541.X. ISSN 0960-7412. 72 Plant Biochemistry, 3rd ed. Academic Press. 2005 p. 517. ISBN 978-0-12-088391-2. Number of copies of ctDNA per chloroplast. 73 AB Kobayashi T, Takahara M, Mijishima Sai, Kuroiwa H, Sasaki N, Otter N, et al. July 2002. Detection and localization of a chloroplast-encoded Hu-like protein that organizes chloroplast nucleids. The Plant Cell. 14, 7 1579-89. Do. 10.1015/TPC.002717. PMC 150708. PMID 12119376. 74 AB Kolodner R, Tuari KK, January 1979. Inverted repeats in chloroplast DNA from higher plants. Proceedings of the National Academy of Sciences of the United States of America. 76, 141-5. Bibcode, 1979 NAS, 76, 41K. Do. 10.1073/NAS.76.1.41. PMC 382872. PMID 165926112. 75 AB Palmer J.D., Thomson W.F., June 1982. Chloroplast DNA. Rearrangements are more frequent when a large inverted repeat sequence is lost. Cell. 29, 253750 do. 10.1016/0092. 8674, 82, 90170-2. PMID 6288261. S2 SID 11571695. 76 Odahara M, Kishita Y, Sekine Y, August 2017. MSH1 maintains organelle genome stability and genetically interacts with RECA and REC in the Moss-Fiske Mitrella patterns. The Plant Journal. 91. 3455465 do 10.1111/tpj.13573. PMID 28407-383. 77 Rowan B.A., Oldenburg D.J., Bendikirch, June 2010. REC maintains the integrity of chloroplast DNA molecules in Arabidopsis. Journal of Experimental Botany. 61, 10.2575-88. Do 10.1093/JXB/ERC088. PMC 2882256. PMID 20406-785. 78 ABCDEF Krishnan N.M., Ral B.J., May 2009. A comparative approach to elucidate chloroplast genome replication. BMC Genomics. 10, 237 do 10.1471/2164-10.237. PMC 2695485. PMID 19457-260. 79 Heinhorst S., Canon G.C., 1993. DNA replication in chloroplasts. Journal of Cell Science. 104, 19. Do 10.1242/JCS.104.1.1. 80 ABCDEF Bendikirch, July 2004. Circular chloroplast. Chromosomes, the grand illusion. The plant cell. 16, 7 1661-6. Do 10.1105/TPC.160771. PMC 514151. PMID 1523523. 81 Effect of chemical mutagens on nucleotide sequence. Biocyclopedia. Retrieved 24 October 2015. 82 Bernstein H., Bernstein C., July 1973. Circular and branched. Circular concatenates as possible intermediates in bacteriophage T4. DNA replication. Journal of Molecular Biology. 77, 335561. Do 10.1016/0022-2836, 73, 90443-9. PMID 4580-243. 83 AB Alberts B., 2002. Molecular Biology of the Cell, 4 ed. New York, UA, Garland. ISBN 0-8153-4072-9. 84 Huang CY, Alif Ma, Timis JN, March 2003. Direct measurement of the transfer rate of chloroplast DNA into the nucleus. Nature. 422, 6927 72-6. Bibcode, 2003 Nature.422, 72H. Do 10.1038/Nature01435. PMID 125944458. S2 SID 4319507. 85 carat ABC Martin W, Ruyan T, Richley E, Hansen O, Cornelson S, Linz T. et al. September 2002. Evolutionary analysis of Arabidopsis. Cyanobacterial and chloroplast genomes reveals plastide phylogeny and thousands of cyanobacterial genes in the nucleus. Proceedings of the National Academy of Sciences of the United States of America. 99, 1912244651. Bib code 2002 NAS 9912246M. Do 10.1073/NAS.1824329999. PMC 129430. PMID 122181772. 86 carat Smith Dr. Lee R.W., April 2014. A plastide without a genome. Evidence from the non-photosynthetic green algal genus Polytomella. Plant Physiology. 164, 41812-9. Do 10.1104/pp.113.2333718. PMC 3982744. PMID 245632881. 87 carat Archibald J.M., December 2006. Algal Genomics, Exploring the Imprint of Endosymbiosis. Current Biology. 16, 24R1033-5. Bib code 2006 CBio 16R1033. Do 10.1016/J.Cub.2006.11.008. PMID 171749110. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 88 carat Kasavitsky S, Noto, Mokla TC, Hong F, Sashto Martins G. Serpin M, et al. May 2007. Signals from chloroplasts converge to regulate nuclear gene expression. Science. 316, 5825-7159. Bib code 2007 SCI 316715K. Do 10.1146/science.11400516. PMID 173957993. S2 SID 245901639. Bob Grant, 1 April 2009. Communicating with chloroplasts. The Scientist. 89 carat De Vries, Jan, Curtis, Bruce A. Gould, Sven B. Archibald, John M. 10 April 2018. Embryophyte stress signaling evolved in the algal progenitors of land plants. Proceedings of the National Academy of Sciences. 115, 15 E3471 E3480. Bib code 2018 NAS 115 E3471D. Do 10.1073/NAS.1719230115. ISSN 0027-8424. PMC 5899452. PMID 295812886. 90 carat Nishiyama, Tomoki, Seikayama, Hitoshi, De Vries, Jan. Bustman, Henrik, St. Marcus, Dennis, Ulrich, Christian K. Haas. Fabian B. Van der Stritten, Lisa, Becker, Dirk, Lang, Daniel. Vosolsob, Stanislav, Romborts, Stefan, Wilhelmsen, Pakai. Janitzer, Philip, Kern, Ramona, July 2018. The Shara genome. Secondary complexity and implications for plant terrestrialization. Cell 174, 2448464.E24. Do 10.1000.j.cell.2018.06.033. PMID 300074117. S2 SID 206569169. 91 Jal, Chinchin, Wang, Yuani Wan, Chan, Kaiksun, Marchant, D. Blaine, Franks, Peter J. Randall, David, T. SDE. Chen. Guang, Ramesh, Sunita, Fua, Suyin, Chang, Ben, Hills, Adrian. Dai, Fia, Xu, Doria, Gillam, Matthew, the 12th of March, 2019. Evolution of chloroplast retrograde signaling facilitates green plant adaptation to land. Proceedings of the National Academy of Sciences. 116. 11 5015 5020. Bib code 2019 NAS 116.5015Z. Do 10.1033/NAS.1812092116. ISSN 0027-8424. PMC 64214119. PMID 3080418. 92 Dadras, Armin, First Jansen, Janine M. Darienko, Tatiana. Kroner, Dennis, Scholes, Patricia, Sun, Sichi, Herefurth, Cornelia. Riesberg, Tim P. Irisari, Eika, Steinkamp, Rasmus, Hansen, Make. Bustchman, Henrik, Valorous, Oliver, Bros, Gerhard H. Huecker. Ute, the 28th of August, 2023. Environmental gradients reveal stress hubs. Predating plant terrestrialization. Nature Plants. 1419. 1438 Bib code, 2023 Nat PL, 9.1419D. Do 10.1038/S41477-023. 01491-0. ISSN 2055-0278. PMC 105055661. PMID 3764035. 93 Hedgby, Bornati, Wye, August 1997. Mitochondrial and chloroplast phage type RNA polymerases in Arabidopsis. Science 277, 5327 80911 do 10.1126/science.277.5327.809. PMID 9242608. 94 Harris A., Boynton J. E., Gillam N. W., December 1994. Chloroplast ribosomes and protein synthesis. Microbiological Reviews 58, 4754 do 10.1000/mmBr.58.4.700-754.1994. PMC 372988. PMID 78545253. 95 ABCDEFGHIJ Sol J, Schlieffen, March 2004. Protein import into chloroplasts. Nature Reviews Molecular Cell Biology. 5, 3198. 208 do. 10.1038 /NRM 1333. PMID 1499100. S2 SID 32453554. 96 Campbell Na, Rees JB, Yori LA, Kane ML, Wasserman, Mineski. PV, Jackson RB, 2009. Biology, 8th ed. Benjamin Cummings. Pearson. P 340. ISBN 978-0-8053-6844-4. 97 Lung SC, Chuang SD, April 2012. A transit peptide-like sorting signal at the C-terminus directs the Bienacia-Synispicici preprotein receptor TARC159 to the chloroplast outer membrane. The plant cell 24, 1560-78 do. 10.1105/TPC.112.0962448. PMC 3398564. PMID 225173118. 98 Wajeman K, Sol J, March 1996. Phosphorylation of the transit sequence of chloroplast precursor proteins. The Journal of Biological Chemistry. 271, 11 6545-54. Do. 10.1054/JBC.271.11.6545. PMID 8626459. 99 AB Métis, Sol J, January 2000. 14-3-3 proteins form a guidance complex with chloroplast precursor proteins in plants. The plant cell 12, 153-64 do. 10.1005/TPC.12.1.53. PMC 140214. PMID 106349007. 100 Jarvis P, Sol J, December 2001. TARC, TIC, and chloroplast protein import. Biochimica et Biophysica Acta, BBA, Molecular Cell Research. 1541, 1264-79 do. 10.1016/S0167-4889, 01, 00147. 1. PMID 117506663. 101 Wise R, Huber J.K., 2007. The Structure and Function of Plastids. Springer. pp. 32-33. ISBN 978-1-4020-6570-5. 102 Edogonium link X Hearn, 1900, 17. Algae BASE. Retrieved. 19 May, 2013. 103 Chlamydomonas Ehrenberg, 1833, 288. Algae BASE. Retrieved. 19 May, 2013. 104 Spirogyra link, 1820, 5. Algae BASE. Retrieved the 19th of May, 2013. 105 Cyrogonium Kutzing, 1843, 278. Algae BASE. Retrieved the 19th of May, 2013. 106 Zinuma C.Agod, 1817, Xxyi, 98. Algae BASE. Retrieved the 19th of May, 2013. 107 Micrasterias C.Agod X Ralphs, 1848, 68. Algae BASE. Retrieved the 19th of May, 2013. 108 John D. M. Brookage, Witten B.A., 2002. The Freshwater Algal Flora of the British Isles, an identification guide to freshwater and terrestrial algae. Cambridge, Cambridge University Press. p. 335. ISBN 978-0-521-77051-4. 109 Fuxby, Homble F., October 1996. Mechanism of proton permeation through chloroplast lipid membranes. Plant physiology. 112, 275966 10.1104/pp.112.2.759. PMC 158,000. PMID 888337. 110 Joyard J. Blockmar, Dowse R. August 1991. Molecular aspects of plastide envelope biochemistry. European Journal of Biochemistry. 199, 3489509 10.1432/J.1032/1432.1991.TB16148.X. PMID 1868841. 111 Chloroplast. Encyclopedia of Science. Retrieved the 27th of December, 2012. 112 ABCDEFG Campbell Na, Rees JB, Yori LA, Kane ML. Wasserman, Mineski PV, Jackson RB, 2009. Biology, 8th ed. Benjamin Cummings, Pearson. pp. 196197. ISBN 978-0-8053. 6844-4. 113 Koik H., Yosho M., Kashino Y., Sato K., May 1998. Polypeptide. Composition of envelopes of spinach chloroplasts, two major proteins. Occupy 90% of outer envelope membranes. Plant and cell physiology. 39, 552632. Do. 10.1093/oxfordjournals.pcp.0294000. PMID 9664716. 114 Cola RH, Hansen Mr., January 2000. Plastide tubules of higher plants are tissue-specific and developmentally regulated. Journal of Cell Science. 113, pt 181-9 do. 10.1242/JCS.113.1.81. PMID 10591627. Archived from the original on the 20th of September, 2016. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 115 Gray JC, Sullivan J.A., Hibberd J.M., Hansen Mr., 2001. Stromules. Mobile protrusions and interconnections between plastids. Plant Biology 3.322.333. Bibcode 2001 PL Bio 3223G. Do 10.1055 S2001-15204. S2 SID 84474739. 116 Scott at M, Barton K, Bordish B, Klosgen RB, Mather J, April 2011. Plastide stromule branching coincides with contiguous endoplasmic reticulum dynamics. Plant physiology 155, 4. 1667-77. Do 10.1104/pp.110.17048. PMC 3091094. PMID 21273446. 117 Scott at MH, Griffiths S, Mather N, Barton K, Wozni Mr., Dunn N. It al. April 2012. Differential coloring reveals that plastids do not form networks for exchanging macromolecules. The plant cell 24. For 1465-77 do 10.1105/TPC.111.0953998. PMC 3398557. PMID 22474180. 118 Brunkard Joe, Runculum, Zambrisky PC, August 2015. Chloroplasts extend stromules independently and in response to internal redox signals. Proceedings of the National Academy of Sciences of the United States of America. 112, 32100449. Bibcode 2015 NAS 11210044B. Do 10.1033/NAS.15115701112. PMC 4538653. PMID 26150490. 119 ABCDEFGHIJ Burgess J, 1989. An Introduction to Plant Cell Development, PBKED. Cambridge, Cambridge University Press. P46. ISBN 0-521-31611-1. 120 Plant peptidoglycan precursor biosynthesis, conservation between moss chloroplasts and gram-negative bacteria. 121 AB Whatley J.M., 5 July 1994. The occurrence of a peripheral reticulum in plastids of the Gymnosperm Welwitschia mirabilis. New Phytologist. 74, 2215222, 10.1111/J.1469. 8175.TB02608.X. 122 ABC Wise R, 2007. The Structure and Function of Plastids. Springer. pp. 17-18. ISBN 978-1-4020-6570-5. 123 Manuel L. Quisp J. Mayfield SP, August 2007. Structure of the Chloroplast ribosome, Novel Domains for Translation Regulation. PLOS Biology. 5, 8E209 10.1031/journal.bio.0050209. PMC 1939882. PMID 176831999. 124 AB Bry, P. Lebengut, M. Sora, M. Boehringer, D. Ban, N. 15. February 2017. The complete structure of the chloroplast 70S. Ribosome in complex with translation factor PY. The EMBO. Journal. 36, 4475486 10.15252/emj.2016959959. PMC 5694952. PMID 28007896. 125 AB Lim K, Kobayashi I, Nakai K, July 2014. Alterations in RNA. mRNA interaction during plastide evolution. Molecular biology and evolution. 31, 1728-42. 10.1093/Molbev/MSU120. PMID 247105116. 126 Heroes T. Sugiura M. January 2004. Functional Schindalgano. Like sequences for translational initiation of chloroplast. Plant and Cell Physiology. 45, 11147 10.1093/PCP/PCH002. PMID 147494993. S2 SID 10774032. 127 Mar J. Campbell, Carlin S., October 2002. Correlations between Schindalgano sequences and gene features such as predicted expression levels and operon structures. Journal of Bacteriology. 184, 25733-45 10.1018/JB.184.20.5745.2002. PMC 139613. PMID 1227082. 128 Lim K, Futa Y, Kobayashi I, October 2012. Large variations in bacterial ribosomal RNA genes. Molecular biology and evolution. 29, 10.2937-48 10.1093/Molbev/MSS101. PMC 3457768. PMID 224464745. 129 ABCDEFGHIJ Austin Jr., Frosty, Viday Pa, Kessler F., Sterlin LA. July 2006. Plastoglobules are lipoprotein subcompartments of the chloroplast that are permanently coupled to thylakoid membranes and contain biosynthetic enzymes. The plant cell. 18, 71693. 703 10.1105.TPC.105.0398959. PMC 1488921. PMID 167315886. 130 AB Crumpton Taylor M. Grandison S. PNG Kilometer, Bushby Smith. February 2012. Control of starch granule numbers in Arabidopsis chloroplasts. Plant Physiology. 158, 290516. Do. 10.1001/pp.111.1866957. PMC 3271777. PMID 2213540. 131 Zeman SC, Delat T, Messaligi, Umhang M, Stetler M, Metler T, Streb S, Reinhold H, Cotting O, 2007. Starch breakdown, recent discoveries suggest distinct pathways and novel mechanisms. Functional plant biology. 34, 646573 do. 10.1071/FP06313. PMID 3268935. S2 SID 159954416. 132 Roche EJD, 1998. The Molecular Biology of Chloroplasts and Mitochondria in Chlamydomonas. Dordrecht, UA, Chlorocad. Publ. pp. 55565. ISBN 978-0-7923-5174-0. 133 ABCDEF Gunning B, Steer MW, 1996. Plant Cell Biology. Structure and Function. Boston, Mass., Jones and Bartlett. Publishers. P24. ISBN 0-86720-504-0. 134 Hansen D., Andrews T.J., Badger Mr., 2002. Variability of the pyrenoid-based CO2 concentrating mechanism in hornworts. Anthoceratophyta. Functional Plant Biology. 29, 340716. Do. 10.1071/pp01210. PMID 3268945. 135 AB Mar Y, Pollock SV, Zial Y, Kunyazami K, Morani JV, June 2011. Identification of a novel gene, CIA6, required for normal pyrenoid formation in Chlamydomonas reinhartii. Plant Physiology. 156, 288496 do. 10.1104/pp.111.17392. PMC 3177283. PMID 215274223. 136 AB Ritalik B, Butler Road, January 1970. The Development and Structure of Pyrenoids in Bulbochelioensis. Journal of Cell Science. 6, 122941 do. 10.1042/JCS.6.1.229. PMID 5417694. 137 Brown Mr., Arnott H.J., 1970. Structure and Function of the Algal Pyrenoid, PDF. Journal of Pharmacology. 6, 1422. Do. 10.1529-8529-1817.1970.tb02350.x. S2 SID 856044422. Archived from the original, PDF, on 31 May 2013. Retrieved 31 December 2012. 138 A B C D E F G Bussy Y, Shimoni E, Wiener Kapong R, Charu VD. Nevo R, Ifrati E, Reek Z, 2019. Fundamental Helical Geometry. Consolidates the plant photosynthetic membrane. Proc National Acad. SCI USA. 116, 442236622375. Bib code 2019 NAS 11622366B. Do 10.1073/NAS.1905994116. PMC 6825288. PMID 316113887. 139 Infunga S, Biscoff S, Hildbrunner, Agn B, Baginski S, Kessler. F, March 2011. The chloroplast import receptor TARC90 partially restores the accumulation of TARC159 client proteins in the Arabidopsis thaliana PPI2 mutant, PDF. Molecular plant. 4, 2. 25263. Do. 10.1093/MP/SSQ071. PMID 21220583. 140 thylakoid. Merriam-Webster Dictionary. Merriam-Webster. Retrieved 19 May 2013. 141 A B C D E F G H Mustardy L, Buttle K, Steinbach G, Garab G, October 2008. The three-dimensional network of the thylakoid membranes. In plants, quasi-helical model of the granum stroma assembly. The plant cell. 20, 10.2552 7 do. 10.1105/TPC.108.059147. PMC 2590735. PMID 1895278. 142 Paulillo Jr., DJ, 1970. The three-dimensional arrangement of intergranule lamellae in chloroplasts. J-cell SCI. 6, 124355. Do. 10.1242/JCS.6.1.243. PMID 5417695. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 143 Shimoni E, Ravhano, O Hardai, Brumfeld V, Reek Z, 2005. Three-dimensional organization of higher plant chloroplast thylakoid membranes revealed by electron tomography. Plant cell. 17, 9. 2586. Do. 10.1015/TPC.105.035035030. PMC 119746. PMID 1605560. 144 A.B. Austin Jr., Ster Elin LA, April 2011. 3-dimensional. Architecture of grana and stromathylacoids of higher plants as determined by electron tomography. Plant physiology. 155, 4. 160111. Do. 10.1104/pp.110.170647. PMC 3091084. PMID 212243441. 145 chloroplast in a plant cell. Tumegi/science photo. Library. Retrieved 19 August 2020. 146 ABC Campbell Rees JB, Yori LA, Kane ML, Wasserman. Mineski PV, Jackson RB, 2009. Biology, 8th ed. Benjamin. Cummings, Pearson. pp. 191-93. ISBN 978-0-8053-6844-4. 147 Australian scientists discover first new chlorophyll in 60 years. University of Sydney. 20th August 2010. 148 ABC Takaichi S. 15th June 2011. Carotenoids in algae. Distributions, biosyntheses and functions. Marine drugs. 9, 6. 110118. Do. 10.3394/MD906101. PMC 3131562. PMID 217477749. 149 Shapley D. 15th October 2012. Why do leaves change color in fall? News articles. Retrieved 21 May 2013. 150 AB Howe CJ, Barbrook AC, Nisbet Re, Lockhart PJ, Larkham AW. August 2008. The Origin of Plastids. Philosophical Transactions. Of the Royal Society of London. Series B, Biological Sciences. 363. 1504 2675 85 do. 10.1098/rstb.2008.0050. PMC 2606771. PMID 184688982. 151 Introduction to the Rhodophyta. University of California. Museum of Paleontology. Retrieved 20th May 2013. 152 ABC DEFG Campbell Rees JB, Yori LA, Kane ML. Wasserman, Mineski PV, Jackson RB, 2009. Biology, 8th ed. Benjamin Cummings, Pearson. pp. 200-201. ISBN 978-0-8053. 6844-4. 153 Lawton Jr., March 1988. Ultrastructure of chloroplast. Membranes in leaves of maize and ryegrass as revealed by selective staining methods. New Phytologist. 108, 3277283. Do. 10.111469-8137.1988.tb 04163.x. JSTA 24339294. PMID 33873933. 154 Roberts K. 2007. Handbook of Plant Science. Chichester, West Sussex, England, Wiley. p16. ISBN 978-0-470-05723-0. 155 Campbell-Na, Rees J.B., Yoy L.A., Kane M.L., Wasserman, Mineski. P.V., Jackson R.B., 2009. Biology, 8th ed. Benjamin Cummings. Pearson. p. 742. ISBN 978-0-8053-6844-4. 156 A. B. Lawson T. and J. I. L. Morrison. Essay 10.1 Guard Cell. Photosynthesis. Plant Physiology and Development, 6th edition. 1. 157 A. B. Wells, C. Baelish, E. 1979. The mitogenic activity of lipopolysaccharide for spleen cells from germ-free, conventional, and not abiotic rats. Canadian Journal of Microbiology. 25, 9 1087-93. Do. 10.1139/M79-166. PMID 540263. 158 A. B. C. D. E. F. G. H. I. J. K. L. Glyn J. M. Mijishimasai, Yoda D. W. Osteriaun Kilowatt, Vither S. May 2007. Chloroplast Division. Traffic. 8, 545161 do. 10.1011/J.100600-0854.2007.00545.x. PMID 17451550. S2 SID 28088844. 159 A. B. Dawn X.J., Nagaya, Takage I.S., 1998. Microfilaments anchor. Chloroplasts along the outer periclinal wall in Vallisneria epidermal cells through cooperation of PFR and photosynthesis. Plant and Cell Physiology. 39, 12 1299-306. Do. 10.1093/oxfordjournals.pcp.0293334. 160 ABC Takage I.S., June 2003. Actin-based photoorientation. Movement of chloroplasts in plant cells. The Journal of Experimental Biology. 206, pt 12/1963 do. 10.1242/Jeb.00215. PMID 127562777. 161 Zyger E., Talbot L.D., Freekiller S., Srivastavara, Chu J., 2002. The Guard Cell Chloroplast, A Perspective for the 21st Century. New Phytologist. 153, 34154424 do. 10.1046/J.0028. 646x.2001.nph328.doc.x. PMID 33863211. 162 ABCDEFG Padmanabhan MS, Dinesh Kumar SP, November 2010. All Hands on Deck, the role of chloroplasts, endoplasmic reticulum, and the nucleus in driving plant innate immunity. Molecular plant microbe interactions. 23, 11 1368 80. Do. 10.1094/MPMI 05-10-0113. PMID 209233448. 163 Cat Sir L. Chung H.S., Kuaj, How Ga, August 2008. Jasmonate. Signaling, a conserved mechanism of hormone sensing. Current. Opinion in Plant Biology. 11, 442835. Bibcode, 2008 COP, 11428K. Do. 10.1016/J.B.2008.05.004. PMC 2560989. PMID 185838180. 164 Schnur J.A., Shockey J.M., De Boer G.J., Browse J.A., August 2002. Fatty acid export from the chloroplast. Molecular characterization of a major plastidyl acylcoenzyme synthetus from Arabidopsis. Plant physiology. 129, for 1709 do. 10.1104/pp.0032551. PMC 166758. PMID 121778483. 165 ABCD Biology, Concepts and Connections. Pearson. 2009. pp. 108118. 166 Campbell Na, Williamson B., Hayden R.J., 2006. Biology, Exploring Life. Boston, Massachusetts, Pearson Prentice Hall. ISBN 978-0. 13-250882-7. Page needed. 167 Jagendorf at, Uribe, January 1966. ATP formation caused by acid-base transition of spinach chloroplasts. Proceedings of the National Academy of Sciences of the United States of America. 55. 1177. Bibcode, 1966 NAS, 55170J. Do. 10.1073/NAS.55.1.170. PMC 285771. PMID 5220864. 168 AB Bird JM, Timochko JL, Stryer L, 2002. Biochemistry, 5 ed. 4 print. ed. New York, NY, UA, W. H. Freeman. pp. Section 19.4. ISBN 0-7167-3051-0. 169 AB Bird JM, Timochko JL, Stryer L, 2002. Biochemistry, 5 ed. 4 print. ed. New York, NY, UA, W. H. Freeman. pp. Section 20.1. ISBN 0-7167-3051-0. 170 ABC Wampel R. L., Davis R. W., September 1983. Effect of flooding on starch accumulation in chloroplasts of sunflower, Helianthus. A new cell. Plant Physiology. 73, 11958. Do. 10.1104/pp.73.1.195. PMC 10664435. PMID 16663176. 171 Cameo, Shima I, 1979. Starch accumulation and photosynthetic activity in primary leaves of bean, Phaseolus. Vulgaris L. Annals of Botany. 44, 44798484. Do. 10.1093/oxfordjournals.aab.085756. 172 Bird J.M., Timochko J.L., Stryer L., 2002. Biochemistry, 5th ed. W. H. Freeman. pp. Section 19.4. Retrieved 30 October, 2012. 173 ABC Hauser M., Eichelman H., Aha V., Heber U., Lesko, July 1995. Stimulation by light of rapid pH regulation in the chloroplast. Stroma in vivo as indicated by CO2 solubilization in leaves. Plant Physiology. 108, 3 1059 1066 do. 10.1104/pp.108.3.1059. PMC 157457. PMID 12228527. 174 AB Wooden K. Helt H.W., My Love and Sev M., August 1975. The role of pH in the regulation of carbon fixation in the chloroplast stroma. Studies on CO2 fixation in the light and dark. Biochimica IT. Biophysica Acta, BBA, Bioenergetics. 396, 227692. Do. 10.1016/0005-2728, 75, 90041-9. PMID 239746. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF. 175 ABCDEFG Burgess J., 1989. An Introduction to Plant Cell Development. Cambridge, Cambridge University Press. p56. ISBN 0-521-31611-1. 176 Ferro M., Salvi D., Riviere Roland H., Vermat T., Saint-Neurin Bernie D. Grunwald D., et al. August 2002. Integral membrane proteins of the chloroplast envelope, identification and subcellular localization of new transporters. Proceedings of the National Academy of Sciences of the United States of America. 99, 17-1148792. Bibcode, 2002 NAS, 9911487F. Do. 10.1073/NAS.172390399. PMC 123283. PMID 121774442. 177 AB Roland N., Drew M., Dowse R., March 1992. Subcellular distribution of O-acetylserine, thiol, lysine cauliflower, brassica. Oleracea L. Inflorescence. Plant Physiology. 98, 392735. Do. 10.1104/pp.98.3.927. PMC 10802889. PMID 16668766. 178 Ravanel S. Gakier B. Job D. Dowse R. June 1998. The specific features of methionine biosynthesis and metabolism in plants. Proceedings of the National Academy of Sciences of the United States of America. 95, 13 7805 12. Bib code, 1998 NAS, 95.7805 R. Do. 10.1073//nas.95.13.7805. PMC 22764. PMID 9636232. 179 ABCDE Buchanan BB, Gruisim W, Jones RL, eds. 2015. Biochemistry and Molecular Biology of Plants. Wiley Blackwell. 180 Joy R.J., Ferro M., Massillon C., Sainer Bernie D., Salvi D., Garin J., Roland N., April 2010. Chloroplast Proteomics highlights the subcellular compartmentation of lipid metabolism. Progress in lipid research. 49, 212858 do. 10.1016/j.plepras.2009.10.003. PMID 19879895. 181 Proudlove Mo, Thurman the, 1981. The uptake of 2-oxyglutarate and pyruvate by isolated P-chloroplasts. New phytologist. 88. 2255264 do. 10.1469/J.1469-8139-81.TB01722.x. 182 Bal X, Fock M, Pollard M, Olrog J 2000. Understanding in vivo carbon precursor supply for fatty acid synthesis in leaf tissue. Plant Journal 22, 3950. 183 Olrog J, Browse J, 1995. Lipid biosynthesis. The plant cell. 7, 795797 10.1105/TPC.7.7.957. PMC 160893. PMID 764058. 184 Gunning B, Steer MW, 1996. Plant Cell Biology, Structure and Function. Boston, Mass., Jones and Bartlett Publishers. p20. ISBN 0-86720-504-0. 185 ABCDE Burgess J., 1989. An Introduction to Plant Cell Development, PBKED. Cambridge, Cambridge University Press. pp. 54-55. ISBN 0-521-31611-1. 186 Burgess J., 1989. An Introduction to Plant Cell Development. PBKED. Cambridge, Cambridge University Press. p. 57. ISBN 0-521-31611-1. 187 A.B. Possingham J.V., Rose R.J., 1976. Chloroplast replication and chloroplast DNA synthesis in spinach leaves. Proceedings of the Royal Society B. Biological Sciences. 193, 1112-295305. Bib code 1976 RSPSB.193295P. Do 10.1098/RSPB.1976.0047. S2 SID 2691108. 188 Cannon GC, Heinhorst S., 1 January 1993. DNA replication in chloroplasts. Journal of Cell Science. 104, 119. Site CX 10.1.1.1026.3732. Do 10.1242/JCS.104.1.1. 189 ABCDEFGH Imagishima Nishida K, Mori T, Matsuzaki M. Higashiyama T, Kuroiwa H, Kuroiwa T, March 2003. A plant-specific dynamin-related protein forms a ring at the chloroplast division. Site: the plant cell. 15. 365565 do 10.1105/TPC.009373. PMC 150020. PMID 1261599. 190 AB Hashimoto H, Possingham J.V., April 1989. Effect of light on the chloroplast division cycle and DNA synthesis in cultured leaf. Discs of spinach. Plant Physiology. 89, 41178-83. Do 10.1104/pp.89.4.1178. PMC 10559993. PMID 1666681. 191 ABCD Hansen AK, Escobar LK, Gilbert Le, Jansen RK, January 2007. Paternal, maternal, and biparental inheritance of the chloroplast genome in Passiflora, Passifloraceae. Implications for phylogenetic studies. American Journal of Botany. 94, 142-6. Do 10.3032/HP.94.1.42. PMID 21642206. 192 Berkey CW, December 1995. Uniparental inheritance of mitochondrial and chloroplast genes, mechanisms and evolution. Proceedings of the National Academy of Sciences of the United States of America. 92, 25113318. Bib code 1995 NAS 9211331B. Do 10.1073/NAS.92.25.11331. PMC 40394. PMID 8524780. 193 Powell W, Morgan M, McDevitt R, Vendraman G.G., Rafalski J.A. August 1995. Polymorphic simple sequence repeat regions in chloroplast genomes, Applications to the population genetics of pines. Proceedings of the National Academy of Sciences of the United States of America. 92, 17 7759 63. Bibcode, 1995 NAS, 92.7759 p. Do. 10.1073/NAS.92.17.7759. PMC 41225. PMID 764491. In the pines, the chloroplast genome is transmitted through pollen. 194 Stigman S., Hartman S., Ruff S., Boca, July 2003. High frequency gene transfer from the chloroplast genome to the nucleus. Proceedings of the National Academy of Sciences of the United States of America. 100, 15828 33. Bibcode, 2003 NAS 100.8828 S. Do. 10.1073/NAS.14309241000. PMC 166398. PMID 128178081. 195 AB Ruff S., Kacha D., Boca, April 2007. Determining the transgene containment level provided by chloroplast transformation. Proceedings of the National Academy of Sciences of the United States of America. 104, 17 6998 7002. Bibcode, 2007 NAS 104.6998 R. Do. 10.1073/NAS.0700008104. PMC 1849964. PMID 174200459. External links. Chloroplast Cell-Centered Database. Clegg Mount, Gort B.S., Learn G.H., Morton B.R., July 1994. Rates and Patterns of Chloroplast DNA Evolution. Proceedings of the National Academy of Sciences of the United States of America. 91, 15 6795 801. Bibcode, 1994 NAS, 91.6795 C. Do. 10.1073/NAS.91.15.6795. PMC 44285. PMID 8041699. Co-extra research on chloroplast transformation. NCBI Full Chloroplast Genome. Categories: photosynthesis, plastids, endosymbiotic events. Explore our developer-friendly HTML to PDF API printed using PDF Crowd HTML to PDF.